So welcome everybody. Thanks for joining this CNS workshop on brain states. We are pleased to have a great panel of speakers with Tatiana Engel, Anna Levina, Enzo Tagliasucchi, and Valentin Dragoi. Here you can see the schedule of the workshop. Well, my name is Adrian Ponce. I'm a postdoc in Pompeu Fabra University of Barcelona, and the other organizer is Gabriela Mochol. She is at the Autonomous University of Barcelona. So the idea of the workshop was to discuss brain states at different spatiotemporal scales. And for this, we have invited researchers such as Enzo Tagliasucchi, who is professor at the University of Buenos Aires, and he's an expert on critical dynamics. He studies spatiotemporal patterns of fMRI during resting state, unconscious state, and states perturbed by psychedelics. And we also have great pleasure to get Anna Levina. She's one of the most important researchers in studying self-organized criticality. In particular, the study of neuronal avalanches, how they self-organize in neural networks, and what are the functional consequences of these special patterns. And maybe Gabriela wants to present the two speakers.
So two other speakers we invited. It's Valentin Dragoi. He's assistant professor in McGovern Medical School at the Texas University. And in his group, he studied how the perception and cognition emerge from the population activity of neurons in cerebral cortex using, in my opinion, cutting-edge experimental technologies. And the other speaker is Tatiana Engel. She's assistant professor in Cold Spring Harbor. She also studied how the perception and cognition emerges from the pattern of cortical activity, but using slightly different techniques, meaning mathematical modeling and computational neuroscience methods. So we will start with Tatiana, but before we start, I just wanted to emphasize that we will, after each talk, the talk is planned around 30-35 minutes. After each talk, we have a short break to have questions, but if you will not be able to ask this question after the talk, after the 4 talks, we will have panel discussion of 25 minutes, and we will try to address all the questions which we couldn't during the talks. So if you have any questions, you can raise the hand in the emoticon panel of Zoom or post it on chat, and we will on the Zoom chat, and we will follow it, and we'll try to do our best to address all the questions during this presentation. So I give the floor to Tatiana. Welcome, and many thanks for being here.
Yeah, thank you so much for the opportunity to speak in this very exciting workshop, and thank you everyone who came to listen to my talk. I would like to start today, or I would like to present you today our recent work trying to understand what is a brain state on the global scale of the brain. And I guess usually when we think about brain state, we think about transition from wakefulness to sleep. And obviously this is the most parsimonious change in brain activity which happens on the transition from waking to sleep. But even during wakefulness, I guess now we recognize that brain exhibits very rich spatiotemporal dynamics, and we are just beginning to understand what these dynamics mean and how they define the state of the brain. I would like to start by showing you this video of spontaneous neural activity in the human brain recorded with fMRI. And you can appreciate that in this subject who is not engaged in any specific behavioral task, who is awake and just resting in the scanner, you can see these rich patterns of activity which clearly have a lot of structure. And I guess now all of us understand that these dynamics, which are the characteristics of varying brain states during wakefulness, they will influence how the brain will respond to sensory stimuli, how it will perceive, and how it will act. So how do these dynamics arise? To study this, animals model provide a better handle because, for example, in the mouse, we can have access nowadays to both. We can measure neural activity on the global scale, similar to what is measured in the fMRI. But in this video, I show you data recorded by Simon Mochol in Anne Churchland's lab, and you see activity across the mouse dorsal cortex recorded with wide field calcium imaging. So in this recording, in this mouse, neurons express calcium indicators, and we directly measure calcium signals across the entire dorsal surface of the cortex. And just as you saw in the human fMRI, in this mouse, which is awake and behaving, you see a lot of ongoing dynamics, which are spreading through the entire cortex and appear to have very complex patterns. But the advantage of the mouse model is that we also have very detailed data on anatomical connectivity. In particular, here I show you a map of long-range cortical connectivity, which was compiled by Allen Institute using tracer injections. So these data show us how each brain area connects to another brain area. And clearly, like, it has been long hypothesized that large-scale dynamics arise from this connectivity structure. But so far, we don't have good mathematical models which can tell us how this rich spatiotemporal dynamics, which defines the global state of the brain, arise from the anatomical connectivity patterns. This will be the focus of my talk today. The work which I will present was performed by Yan Liang-Shi, who is a postdoc in my lab, and he developed a computational framework which is able to integrate anatomical connectivity data of the mouse cortex with the large-scale mesoscopic functional activity measured with wide field imaging to develop a mathematical model of cortical dynamics. So where we started with this project is to analyze the data from Anne Churchland's lab, which I already showed you. In this experiment, mouse was engaged in decision-making behavior. It was seeing stimuli on the screen and also receiving auditory stimuli, and it was required to look to the left or to the right, depending on which stimulus was shown. So mouse was engaged in decision-making task, and you can see a video of mouse performing this type of task. And the activity which Simon recorded throughout the mouse cortex, you already saw this video, is very, very rich and widespread. If you're looking at this activity, you would not be able to easily tell that mouse is performing decision-making behavior, because on individual trials, there are very large amplitude fluctuations, which do not necessarily clearly align with the time course of behavioral task. If we would align activity to task-relevant events and average the activity over many, many trials, then you would see the expected patterns in the respective cortical areas. For example, if you pay attention here, if we align activity, if the average activity align to the visual stimulus onset, and average activity over multiple trials, we see activation in the visual cortices. Or in the beginning of the trial, when mouse is grabbing the handles to initiate the trial, we see somatosensory activity in the somatosensory cortex. However, these activations evident in trial average data are not obvious on individual trials, where fluctuations appear to the most dominant mode of dynamics, and the task-related activity rides on it just as a small ripple. To be able to understand how this ongoing activity arises in the cortex, we decided to combine this data with anatomical connectivity data. We used a common coordinate framework from the Allen Institute to align cortical recordings with the connectivity data. Here you see the map of mouse cortical areas from the Allen Institute. And it just shows you a spatial location of visual cortices, somatosensory cortices, motor cortices, and so on in the mouse cortex. Allen Institute also gives us access to the anatomical connectivity between all those areas. Anatomical connectivity data, you can think about it as a matrix, where for each source area, we have a connection strength to each target area in the same and in the contralateral hemisphere. If you just look in this connectivity matrix, just by eye, we can already appreciate there is quite a bit of structure. This matrix is not completely random. For example, I highlighted here the blocks, which correspond to visual systems, somatosensory system, retrosplenial system, and so on. And you can see that within each of those systems, the connectivity seems to be stronger than the off-diagonal entries. But there are also entries which don't follow in this pattern. For example, this block of strong connections, we don't know at this point whether those connections are important, but clearly they are present in the connectivity. So what we did, we coarse-grained the wide field imaging data according to the map of cortical areas from Allen Institute, and we average activity within each cortical area to get data which looks like this. So now we have a time series of activity in each of those areas across the mouse cortex during behavior. You can also think about this data as time series of activities or time traces of activities in all of these areas which are recorded simultaneously. So this is a single trial activity, and in this representation, you can also see that activity varies quite a bit across areas. It shows a lot trial-to-trial variability, and it seems it is a tall order to try to predict all these fluctuations in single trials. But this is what we attempted. We reasoned that in the cortex, each area receives inputs from all other cortical areas. So we wondered to what extent activity in each cortical area can be explained by the input it receives from all other cortical areas. To this end, we built a linear model where we predict activity in the target area, I denoted X of t here, by activity in all other cortical areas measured at the same time. And to compute the input which this area will receive from its sources, we used connector weights. So this model has no free parameters. We don't feed this model. We just compute input to an area as activity of source areas weighted by the connector weights. And we also allow by a scalar, which can multiply this activity, which we called input-output gain, because both the connectivity and activity data are in somewhat arbitrary units, and we cannot expect that there will be much in the units after we perform this computation. So here is an example of few areas of what we obtained. This is the actual data measured in a set of cortical areas, and this is prediction of this of the input to this area arriving from all other areas. And I have to mention that in these predictions, we exclude. Is there a question? Okay. If you could mute yourself, that would be great if you don't have a question. So when making this prediction, we exclude the diagonal of the connector matrix, both within the same and contralateral hemisphere to avoid self-prediction. So here we predict activity of one area, but only using activity of all other areas. And already visually, you see quite a good correspondence, and it becomes even more striking if I will put those traces on top of each other. So in blue, you see data, activity data in each of the areas, and in red, you see predictions, which are inputs arriving from other areas, and we can obtain a very precise match. That was quite surprising to us. It suggested that dynamics of cortex are really happening in big part because of recurrent interactions in the cortex. But we wondered to what degree this result may be expected or trivial. If activity is somewhat correlated all over the cortex, maybe we don't need to know the exact anatomical weights between different areas, and any random connectivity matrix would be able to give us the same level of accuracy in prediction. To test for the importance of the specific mouse connectome in making these predictions, we did the following experiment. We used the original connectome, And then we randomly shuffled the rows and columns of this matrix within each hemisphere. This is how the shuffled connectivity would look like. The overall strength of the connectome weights is the same, but their ordering is disturbed. So now we make the same prediction using the shuffled connectome. And by doing many shuffles, we obtain the average R-square prediction, which would arise from just a random shuffled connectivity. And red line shows here the original prediction, the original R-squared, which we got with the original connectome. And as we can see, if we use the true structure of connectome, the prediction accuracy is substantially higher, which indicated to us that the anatomical connection weights are actually important to define activity in each of the areas. We were quite surprised by this result because we combined activity data recorded in one set of mice in one laboratory and connectivity data, which was obtained in completely different institutions from very different mice. So if this relationship is true between activity and connectivity, we reason it should hold across many behaviors and many different experimental contexts. To test this idea, we used another data set, which was also a wide field imaging recording performed by Nick Steinmetz back in the Carandini lab at UCL. And in this data set, the mouse was not engaged in any specific behavior. This is what we call spontaneous activity. Mouse is just resting under the microscope, and maybe it's performing spontaneous movements such as whisking or twitching. In this data set of spontaneous activity, we also found a remarkable correspondence between activity in each cortical area and input this area receives from all its cortical targets. So when we had this result, we wondered to what extent this prediction depends on knowing activity all over the cortex, right? So now we predict activity of one area using activity of all other areas. Are all these links and connections in the cortex are equally important? Or maybe there are some dominant links which define the dynamics across the cortex. To answer this question, we analyze the composition of inputs which each area receives from its sources. So what you see here, it's an example, it's just one row of the connectivity matrix. This is connection strength to area SSPLL from all other cortical areas, and the height of the bar indicates the strength of the connection. But the reason maybe we should not look just in the connection strength, but in the effective input which each area receives. So we weighted this input vector by the average activity in each of the source areas. And in most cases, this weighted connectome or effective connectome agrees with the original, but sometimes there are differences. For example, here you can see that although SSPLL receives strong input from auditory cortex, because auditory cortex has relatively low activity, then effective connectivity would be bigger. So what we did next is to examine this composition of inputs received by each area and trying to draw a graph to represent the most significant connections in these interactions. We thresholded those graphs by selecting a threshold, and we dropped all weak connections, and we kept only strong connections. And you can see here that for most areas, there was just a few inputs which would dominate activity of that area. For example, if you look in this motor area MO, it has one strong input from area FRP, frontal pole, and all other inputs are very weak. So when we would threshold this input vector, we would draw a single arrow from frontal pole to area MO, and there will be no further connections. So what we did after thresholding the effective connectivity matrix, we would only keep strong connections and draw arrows between areas in the place where we have strong connections. Then we would get a graph, which we would call a cortical constellation. And in this graph, there will be some special areas which will not get any inputs. This is, in this particular case, is a frontal pole. You see that if it receives very distributed inputs from very many areas. And there is not a single input which would clearly predict frontal pole activity. And therefore, after thresholding, this area will be just standing by itself. So we call such areas the main stars of cortical constellations, or we also call them the driver areas. And the rest of the areas, which can be clearly predicted by just few inputs, we call them driven areas. So after we did this analysis, starting the analysis at different cortical area, we identified 6 cortical constellations, these 6 subnetworks, which corresponded to the motor system, somatosensory system, olfactory system, auditory system, visual system, and retrosplenial system. And in this constellation, we had a few main stars highlighted here with circles. These are areas whose activity cannot be predicted by a few inputs, but arises from a large combination of small inputs. So this indicated to us that not all connections in the cortex are equally important to explain dynamics, that there are few strong connections which clearly dominate input to most of the areas, while there are few areas which cannot be easily predicted, which suggested to us that these dynamics are actually relatively low dimensional, and we decided to build a model of these dynamics. And as suggested by our analysis of cortical constellations, we come up with the following model of network dynamics. We divided all cortical areas in driver areas, these are the main stars of our constellations, and driven areas, these are areas which can be predicted by just by the few inputs. And then we reason that we cannot predict activity of driver areas, so those activities of these 6 areas, we will take it from data, from the recording, and we will try to predict activity only in driven areas. And in this model, there is again no free parameters. We will use anatomical connectivity weights from driver areas to driven areas, which we treat as effectively feed-forward connectivity. And the recurrent connectivity between driven areas also comes from the connector. So in mathematical form, this is how our model looks like. We had a vector of activity in all cortical areas, which we now divide in a set of driver areas denoted by A and driven areas denoted by X. And we will use activity of driver areas to predict activity of driven areas. And in this model, we divide the connectivity matrix in effectively feed-forward part. This is inputs from driver to driven areas, as well as recurrent connectivity between driven areas. So effectively, we would get the following dynamical equations, where I is the input from driver areas, and X is activity of driven areas, which we are trying to predict. So in this model, the only aspect which we don't know is the time scale of recurrent interactions. So we get data as in the calcium imaging and the specific imaging frame, but we don't know what is the time scale in this equation and how quickly those recurrent interactions can happen. But it is something which we could test with our model. We could assume that this time scale is very slow or very fast and try to predict activity of driven areas, X of t. And depending on which time scale we assume, we can see how it changes the accuracy of our predictions. So the first assumption which we could make is that these recurrent interactions are very slow. In this case, the system would constantly adjust to the changing level of input from driver areas, and recurrent interactions play no role because there is no time for them to unfold. If we use this simple feed-forward model, you can see the prediction of activity in blue from this from the 6-dimensional model would be poor. So there is not a good match between activity measured in each area and predicted by the model in both data sets during decision-making and spontaneous activity. Then we can assume that recurrent interactions happen faster. So there is input arriving in each cortical areas, but between two imaging frames, maybe it is enough time for one loop of recurrent interaction. So the activity across driven areas can loop back one more time to change itself through recurrent interactions. If we assume this little bit faster time scale, we get a better match between measured activity and what is predicted by the model. So if we assume another loop of recurrent interaction, so now activity can loop twice through the recurrent loops of the cortex, we see the prediction becomes even more accurate, and so on. The more recurrent interaction loops we would add to our model, the better becomes the match between predicted and measured activity. This suggested to us that recurrent interactions happen on very fast timescales, faster than what we can measure with calcium imaging. And this suggested us a model architecture where we would account for all infinite number of those recurrent interactions, or you can think about it as that for each level of input, the system, the recurrent dynamics between driven areas would equilibrate to the new fixed point defined by the external input. So we can sum up all these infinite interaction loops to derive an effectively feed-forward equation. So in this equation, we predict the activity of driven areas X from the input arriving from driver areas. In this analytical equation, there are no free parameters, and the effectively feed-forward structure is defined by the anatomical connectivity. But as you can see, this relationship is non-linear because it accounts for both feed-forward input from driver areas, but also the recurrent connectivity, recurrent interactions between driven areas, resulting in this non-linear relationship between activity and connectivity. So let's see how this six-dimensional model, which takes into account all recurrent interactions, can account for cortical dynamics in single trial. And the match was remarkably good. So we got pretty much the same R-squared value, which we would get by taking all input areas into account. So these are predictions of a six-dimensional model overlaid on the measured neural activity data. And across all areas, we would see very good correspondence both during decision-making task and in spontaneous activity. So just to summarize this part, we were able to derive a six-dimensional analytical equation which can predict activity all over the cortex from activity of only 6 driver areas. And this model has no free parameters. All entries in this connectivity matrix are derived from the biological connector. And this result applies to different experimental animals, and it applies during different behavioral contexts, suggesting to us that connectome fundamentally defines the cortical dynamics on the large scale. Also, this analysis showed to us that recurrent interactions happen on relatively fast time scales, so that in calcium imaging, we effectively look in steady state of these dynamics for each changing level of input from driver areas. With this, I move to the last part of the talk, which is about predicting behavioral movements based on large-scale cortical dynamics. And this was motivated by the paper which Simon published himself on this dataset, where they noticed that if they used video data, these spontaneous movements which animal is doing during the task, then this video data can predict quite a bit of variability in neural activity across the cortex. And already in their paper, there is an indication that this neural activity is actually relatively low dimensional. Here you can see how many video dimensions can explain variance in neural activity data. And this graph already indicates that there are only 6 to 8 video dimensions which are important for predicting neural activity. And this number matches the size of the number of the driver areas which we were able to identify with our analysis. Therefore, we hypothesize that this relationship between spontaneous movements and neural activity is actually some representation of this low-dimensional dynamics which we were able to identify with our analysis. And to test this idea, we did the following. We tried to predict video data using linear regression, and we do prediction using different kinds of neural activity. First, we can use full pixels, all pixels in the wide field imaging data. This would be the most high dimensional representation of this activity. It is the same as was used by Simon in his analysis. We could also divide this data into areas, coarse grain this activity, and the average activity within each area to get this areal regional activity and try to use this to predict behavioral movements. Obviously, this step loses some information, so we could expect the predictions would be somewhat weaker. But then we can also use our 6-dimensional model, where we only use activity in 6 cortical areas to predict activity everywhere else in the cortex. And then by using this 6-dimensional model, we can try to predict behavioral movements. And we can ask, how much do we lose when we go from full set of cortical areas to this low-dimensional 6-dimensional model? Here is what we got. So this is a prediction of different dimensions in video motion energy and in video itself using the full set of pixels in the wide field imaging data. This gives you just a baseline of how much can we explain in the video based on neural activity across the cortex. If we use areal activity, the predictions become overall slightly worse because, as I said, we lose some detail in this neural activity, so we can expect that we would explain slightly less variance, but the drop is not significantly big. And finally, if we move on to use our 6-dimensional model, the predictions of this model are on par with the predictions of the aerial activity, or sometimes even better, suggesting that indeed the relationship between behavioral movements and neural activity is low-dimensional, and that likely this mesoscale, large-scale activity doesn't encode fine details about behavioral movements, but more like a behavioral state, which is relatively low-dimensional and captures some patterns of behavioral movements which correspond to specific low-dimensional patterns of neural activity. So this brings me to the end of my talk. What I showed you today is that the set of about 6 driver areas are sufficient to account for the rich cortex-wide dynamics in the mouse. And therefore, our approach is a form of dimensionality reduction. However, our dimensionality reduction is not merely tries to capture variance in the data, such as, for example, principal component analysis would do. Instead, we rely on the biological ground truths in the form of the connectome, which allows us to find an interpretable dimension reduction which universally applies across different animals and behavioral contexts. And I would like to highlight that this is not the case with principal component analysis, for example, because although we would define the same 6-dimensional subspace as the principal components would do, the axis within subspace would vary with principal component from behavior to behavior, from animal to animal, making interpretation of those axes difficult. Instead, relying on connectome, we can find the same set of anatomical areas which can apply across animals and behaviors. And finally, we found that 6-dimensional model of dynamics can predict behavioral movements, suggesting that this relationship between neural activity and movements is a signature of something like a low-dimensional behavioral state. So at this point, I would like to thank everyone in my lab, in particular Yan Liang-Shi, who performed all this work. And I would like to thank our collaborators, Simon Mochol and George Steinmetz, who shared their data with us. And I would also like to thank all our funding sources. And I guess now I'm ready to take questions.
Could I start?
I just noted that I was muted, so thanks a lot for the talk. We have 5 minutes for the questions, so if anyone wants to ask. Yeah, we have 2 participants. So Anna, please.
Milena was before me, but maybe
Okay,
I just
sorry,
very
I-
briefly ask. You use the expansion, including as many loops as possible, or like infinite loops. If you would cut off by say, so how many loops you actually need? You showed somewhere that few loops are not enough, but already like 10, is it full performance, or how many are needed?
So we didn't, I guess, check it this way, right? So after we noticed that adding more and more loops would always improve performance, then we tested the model which would have infinite number of loops, which would give us a very compact analytical expression. We found that we pretty much saturate to the level which we get if we use all cortical areas to make predictions. So we didn't kind of study these saturation curves to the very large number of loops. I think we stopped somewhere at 4. But because including infinite number of loops gave us a good prediction, I guess we think that dynamics are definitely faster than the 30 hertz frame rate which we have in capsule imaging. What exactly the time scale is, I feel we cannot really say with this type of data. So we would require recordings which have the faster time course of neural activity available, such as neural pixels recording maybe, to be able to more precisely define the time scale of recurrent interactions.
Okay, and we can go to Milena question.
Hi. First, thank you for this very interesting talk. And I actually had 2 questions, but one of them is related to Anna's question. So I think I'll start from that one. I noticed that when you found this reduced form for your equation, for the effective feed-forward structure, the feeding of each individual area seems to be very different. Some of them seems to be very well fitted, while others seems to be not as well fitted. And I was wondering if you had considered that different areas would need different levels of recurrent loops, or a different number of recurrent loops to actually represent its activity.
This is definitely true. The prediction is not equally good across all areas. There is a range of kind of R squared. So the numbers which I showed you would be the average R squared. And in part, it is because not all areas are equally well visible in the wide field imaging data. Some of the areas which are in the part of the image where the cortex falls off will have a relatively noisy signal just because how they are situated. For example, if you look in these areas here along the midline of the cortex, the activity there would be just naturally very noisy because the real structure of the cortex falls in and it's not very well visible with the wide field imaging. So this is one contribution to heterogeneity of predictions across the areas. But also, I have to say that this model is somewhat limited right now because we only focus on cortical inputs and we completely ignore the subcortical inputs, which may affect different areas to a different degree. So I guess it would be very interesting in the future, like with other types of data sets, to see to what extent inclusion of these subcortical inputs can improve predictions in areas where the model is not doing so well.
Okay. Okay, so I guess Anna.
If there is still time, I wanted to follow up because
Just 1 minute.
What is an eigenvalues of this matrix? Do you know, like the leading eigenvalue, does it kind of blow up activity or decrease it over time, over iterations of the matrix?
Yeah, we looked at it and I don't remember honestly out of the top of my head. And the prediction which we end up having is non-linear. So it's not like exactly that the eigenvalues of this matrix would predict the dynamics. But yeah, I should double check. I'm not sure at this point. Thank you.
Okay, so we will thank you, Tatiana, very much once again, and I will give a space to Adrian and the next talk of Anna.
Our next speaker is Anna Levina. Maybe you can try sharing your screen, Anna. Yeah, perfect. Well, you can start whenever you want.
Okay, thanks a lot for having me here in this exciting workshop. I would talk, instead of this global states, about more localized states, how we can capture them, and how potentially they can be important for the information processing and computations. So to start with, let's ask, what is the state? And to my knowledge so far, the fair answer would be that we don't know any really good definition yet. We know for some states that can be clearly labeled behaviorally, as also Tatiana mentioned, the sleep stages. On the other side, there are few states that we can label from observing the activity of neurons or population of neurons. For example, a critical state that I would mention in a few seconds. And my personal attempt to answer is that the state would be a low-dimensional, lossy most probably, but computationally relevant description of neural activity. This is very general. However, so far, I think this is the best we could capture all what we think about the states in the brain. And let me introduce a little bit what is the critical state and where it is situated. Let's imagine in a caricature example that we have a very mixed up chaotic system. Here, all the small parts are random and we cannot get any information. On the other side, we could just order all the red pixels to one side, all the orange pixels to another side. This would be an ordered state where also not much interesting is happening. In case we would have some kind of a parameters that allows us to move continuously from chaotic to the ordered side, we would meet the border between these two states, and this border would be a critical point. In many situations, for example, for the pictures, this border would also present Paulo scalings in different observations and the long correlation lengths. On the other side, if we are not interested in going from order to chaos, we could think about more maybe neuroscience relevant property of transmitting the information. And in a very kind of crude border state, the information is not spreading. For example, we have a network and we put in some information, but it's locked. On the other extreme, we have an information that spreads exponentially and takes over the whole network. And somewhere there is a border where statistics of the passes that information parts or the parts of the network the information reaches follows a Paulo scaling. And this would be a critical point defined by the spread of the information. Why is it important to think about it if we want to understand the brain dynamics? One reason for it, found in mainly theoretical studies, that indicated that being close to this critical point or at the critical point can improve dramatically information capabilities and computational power of the networks, which bring by the idea that it would be the perfect state for operation of the brain. And indeed, there were found different signatures of operating close to the critical state in different modalities of recordings, ranging from spikes to EEG over all other possibilities. And additionally, theoretically, we know that it is possible to achieve a self-organization in the network toward this critical state, for example, by synaptic plasticity. Is it a free lunch? We know they're unknown, so there is a price to pay for this optimality. Here, we consider an example presented by Luca Cocchi, with a system which can be tuned either to a red, very subcritical state where information is not spreading, or to the yellow, enormously, vigorously spreading information that overtakes the network very fast, over the blue critical point. If we look at how the activity of the network that is driven by the slow input looks like in the red state, it is a small fluctuations which die out exponentially. If we would look at the supercritical state, the amplitude of activity is very high. However, the variance around this amplitude is still very small. But if we would go to the critical point, we would get a huge variety of responses to the drive, and activity would have a very large fluctuations of the amplitude. It has a positive side. If we would consider the autocorrelation of the activity trace, at the critical point, it would have a largest autocorrelation, which potentially allows the system to integrate the information over very long periods of time. However, at the same time, coefficient of variation is also maximum. So if there is a noise in this information, we would also amplify it, and we would have a huge variance in the readout, which might not be what we want in many real life scenarios. So we have potential optimality, and we have also potential overly noisiness of a closeness to the critical state. So what we could put forward as a hypothesis is that possibly there are different states that would be optimal for different tasks. And maybe locally in the brain, there would be different states in different brain areas. So from the idea that there is this one optimally critical brain, we would go to the thinking about, in a cartoon form, a brain that has different states in different parts and possibly at different times, varying maybe around the critical state, or maybe if we would measure the state in any different way around the wandering around the state space. It was already observed by John Murray that if we would consider states in terms of autocorrelation timescales, they change across the brain hierarchy. And also on the theoretical side, it was found that there is a relationship between simplicity of the task and the optimal state to solve this task. So in general, for me, the main question would be, on one side, how to measure the state, then how the system can self-organize to the state it prefers, and which states would be best matching for which situations. Today, I will leave out the self-organization part and would start discussing measuring the brain state, here particularly in measuring autocorrelation timescales. So let's dive into the autocorrelation. Autocorrelation is a normalized autocovariance, and it is a convolution of a signal with its time-shifted copy. Normally, autocorrelations are decaying exponentially, and the exponent of the decay of decaying exponentially with how far we shifted the time series, and this exponent defines a time scale. If something is decaying exponentially, we can best see its true shape by plotting it in log linear scale, where the autocorrelations normally would look like a straight line as any exponential. The slope of this line would be inversely related with the time scale. And to remind, the connection to critical state would be that if a system approaches a critical point, the autocorrelation timescales increasing to infinity. It seems to be very straightforward to measure timescales if they look just like a slope of the straight line in a log linear form. However, if we would take a data here from many 1 second long trials of artificial process where we beforehand know the time scale, this is the autocorrelation we obtain. We can easily fit a straight line in it, and for a large part of the time lags, it would describe nicely the data. However, in this case, we knew what is the true time scale. It was given by the scale for Ornstein-Uhlenbeck process we simulated. And this is the real time scale, which is very different from what we would find by numerical fit. So first, before defining the states by the autocorrelation timescales, we need to divide the method to not be affected by the bias in a direct fit procedure. And to do so, Roxana Zerati, a PhD student in my lab, together with Tatiana Engel and later also with support of Yang Langxi, devised a method that allows to counteract the bias in the autocorrelation timescale fitting by proposing a well-selected set of generating models. So the main idea is that we would want to get the generating model with the match statistics to the real data, such that all the factors that influence the bias would be matched in our generative model, and then we could directly feed an autocorrelations produced by the generative model with approximate Bayesian computations to the autocorrelation in the data. And what we can see here, it recovers precisely the ground truth autocorrelation timescales in a simple case where we have just single autocorrelation, single time scale, or if we have a time scale and oscillation combined, or also if we have multiple time scales. You can get the code for fitting your own autocorrelations here. So now we know how to get the autocorrelation timescales without bias. We could look at the real data. And the data was provided by our experimental collaborators, Nick Steinmetz, at that time in Terine Moore's lab, and Mark Giselman in Alex Thiele lab. The recordings were performed on the awake and text performing primates, macaque monkeys in the area V4. The task was to find and indicate small changes in the Gabor patterns like the one you see on the screen. And before monkey would need to indicate or find where the task was changing, it was given a cue that was directing towards the location which would most probably have the change. And we distinguish two different cases. When the Q was forwarding to the locations that were not the receptive field of the neurons we record, or to the receptive field. So if we consider all the trials where the attention was directed not towards the place we record from, and we combine all the data, we would obtain this autocorrelation shape. Here I already presented in log linear scale, and we see that there is not a very trivial behavior, which possibly has a multiple time scales. But now let's have a look what's happening if we take together all the trials where monkey was attending towards the receptive field. And we see that in this case, the autocorrelation indicates a visible change between the two different states, attentional states. We fit this autocorrelations with two time scales process, and we could show that the first time scale does not change. The first time scale describes what's happening in the short legs. However, the behavior at the long legs, the slow time scale, increases very significantly where the monkey attends towards the receptive field. Here is just one example, but we could combine the data from 3 different monkeys that we used in our data analysis. And here I show the first time scale comparing conditions of attending towards receptive field or away from receptive field. This is the first time scale. We see that all the data is nicely aligned at the line, slightly shifting up or downwards, indicating that there is no big change. However, if we would look at the second time scale, we see that most of the data lies above the line indicating the same time scale in attention away and attention in condition. And this we could also check with statistical tests showing that there is no significant change for the first time scale, but a significant change for the second time scale. Now we ask ourselves, how can we explain the large changes in a model? To do so, we design a model consisting of the simple units that are interacting with neighbors within a certain radius. The interactions can be captured by 2 parameters. One captures vertical interactions within a column and would result in a self-activation of the unit. This is a purple parameter. And another one captures the horizontal interactions, the recurrent interactions within a network. If we record from such unit for a very long time in the model, we would obtain this purple trace that look very similar to what we've seen in the recordings from the V4 area V4. Here, however, we know the parameters we put into the model, and we could analytically compute the timescales arising from a different part of the model. Namely, we could analytically compute the timescale that arises from just a simple self-excitation of the neurons. On the other side, we could reduce the model by not taking into account the self-excitation and just looking at incoming recurrent connectivity. And here in pink, we see what would be the timescale generated by the recurrent interactions. This already indicates that our timescales look like they are transitioning from the self-excitation timescale in the very short logs to the timescale that is generated by horizontal recurrent inputs in the network. And we could further illustrate it by considering different local interactions. We start with the same image that we had before, where the unit interacts with its nearest neighbors. And then we place the same number of connections, but within a larger and larger radius. If we would take radius 3, we already see that the autocorrelation follows the self-excitation timescale for a larger amount of lags. And if we go to the random connectivity, we get basically only the self-excitation timescale visible. However, if instead of looking at these individual units, we would pull the activity from the whole network, we would observe same timescale every of these cases, which would be related just to the sum of all the incoming connections in the network. Now, we could use a model and check which changes in the recurrent and self-excitation parameters it would predict for the observed changes in autocorrelation timescales. In the recordings, our timescale was around 120, marked by this green arrow. And we blow up into the parameter interval that is the most relevant for the match between the model and the real data. So what is happening is that we are moving from this green ring of the parameters that are matching unattended away condition towards the purple area that is matching the condition of monkeys attending towards the receptive field. And we could indicate 3 major possibilities how the change from the attended away to attended condition could be realized. The first option is to keep the self-excitation parameters unchanged while changing the recurrent connectivity. This is a very tiny change that would result already in rather large changes in the timescales. And this potentially could be realized by top-down inputs. On the other side, we could increase the self-excitation timescale and compensate this increase by the strong reduction of recurrent interactions. This could be achieved by a combined impact of known increasing communication between layers under attention with some neuromodulatory input. And the third opportunity would be to decrease self-excitation but largely increase recurrency. And so far, we don't know any support for this option. But to sum up what we heard so far, we've seen that sample autocorrelation is a biased estimator. And if we use the proper techniques, we could recover true timescales. We also seen that the activity of the local B4 populations unfold across 2 different timescales, one fast and one slow, and the slow timescale is modulated by attention. So it is longer while monkey is attending towards the receptive field, which means also that the network, that underlying network, is closer to the critical state. In the model, we could capture these 2 timescales and show that they are related to the timescale of self-excitation and recurrent connectivity. However, it still remains an open question, which are the exact mechanisms of the attentional modulation of the timescales. Now, we learned a little bit about how we could measure the states locally and how they could change with a change of behavioral states of attention. And we would briefly dive into understanding which states are optimal for which situation. To do so, we will study a simple task that our visual system, for example, can perform pretty well. Namely, if we would be in real Paris and not just in Paris time, we would possibly, after a few hours, see this nice sunset picture. However, if we would photograph this picture with our camera, we would most probably get one of the images shown above. And that's because in this image, we have very bright and very dark parts. And to resolve all of these details, we would need a very high dynamic range, high sensitivity in both very bright and very dark areas. On the other side, we could imagine it in the realm of auditory perception. For example, if we would consider a response, maybe mimicking the neural response to a very quiet whisper somewhere on the other side of the room. For a while, we don't hear anything, and most probably there is no response in our auditory cortex. However, if there is enormously loud sound very close by, it is so strong that most probably we cannot capture the small changes in the intensity, and it saturates our response. In between, we can discriminate sounds by the intensity, which would help us, for example, to predict whether the object, the source of sound, is approaching us or moving away. And it was clearly shown on multiple examples of single cell recordings that this type of response curves are ubiquitous in especially sensory system. How do we characterize these curves? One important parameter is what is the range of stimuli that allows to discriminate them from the output. This is a discriminable interval marked here by delta. If we would take the simple recurrent network that is just parameterized by the single parameter, there is a critical point that we could define mathematically, and at this critical point it was found that the dynamic range of the network is the largest. And the similar observations were done in experiments on the cortical cultures, which were stimulated by the microelectrode array. Now, just a few words about the model we would look at. It has binary neurons which communicate by the stochastic synapses, each of which communicate the probability of activating the postsynaptic partner when the presynaptic neuron is active. And this global parameter that governs the dynamics of the network is the sum of all outgoing probability from a single node M. And depending on this M, there are different statistics of the signal propagation, where for M smaller than 1, activity dies out with a probability 1. For M larger than 1, there is a non-zero probability of activity staying forever in the network. And if M equal 1, on average, every new activation would result in 1 activation in the following time step. And additionally, 1 over 1 minus M defines the autocorrelation timescale of the network, of the whole network together. So now let's look at this dynamic range problem in the network where we feed in some neurons by the input, we read out from another subset. This would be what is happening, for example, in communication between the different stages of signal processing. Now, the question is how long do we have to integrate the input? If we have an infinite amount of time, then we can obtain such an average response curve. We integrate the input for a long time, we can get a single point as what is the output, the average output of the network. So we get the perfect input-output response curve. However, if we would have only very short or just finite amount of time to evaluate the input before we have to give the output, we would have a large variance that is generated by the finite sampling of the input. And here we would have an overlap between the representations of different inputs in the distribution of the outputs. And this overlap would result in a discrimination errors between the similar inputs. So how does the state, how close is the system to critical, plays into this picture? Let's now look at the very strongly subcritical system, and this is the response to one steady state input level. And this is the other one. They are a bit different, and we could describe both of them by their average produced response over a very long period of time. But if we have, instead of the 10 seconds to evaluate this only single second intervals, they would fluctuate a little bit around this mean response, but not too much, resulting in this response distribution. However, if we would take a network that is much closer to critical and design the inputs that would generate the same responses, we would get a much more variable dynamics. And if we would cut again the same parts, we would obtain a much broader distributions with much larger overlap between these two stimuli. Now, we define this dynamic range by how many stimuli we could capture and what is the range in the stimuli that we could discriminate with the 10% error probability. So here we have again the situation with the infinite time. What we see is that the dynamic range increases while control parameters that is also governing how far is the system from critical, approaching the critical state at m equal 1. And the same is happening if we would measure a number of points, a number of different stimuli that the network can discriminate with a given error rate. However, if instead we have very short observation time, we would not have this monotonically increasing function. We would have a maximum at the intermediate, very subcritical values. And if we increase the time of observation, this maximum would slowly shift towards the critical point. This way, we could summarize it in the short windows. The noise produced by the system actually hinders it from using this maximum dynamic range that it would would obtain in an infinite time. And the subcritical system is preferable. And if we could observe the system longer, the more close to critical system we would get. So to summarize what I was talking about today, first of all, very important, if you use time scales, use the unbiased estimator. Second, the activity in V4 has this different two major time scales that define its autocorrelation shape, and that this time scales, that the longer the time scale is changed by the attentional state. It's becoming longer when the monkey is attending to the receptive field. At the same time, in a stochastic input encoding system, the short observation window generates a noise that would be transmitted to the output of the network. And this requires us to define differently dynamic range. Here, what we find is that if we have sufficient amount of time, the most critical network is preferred. But the amount of time we have to make a decision would decide on what time scale is the best for the system to be truthful to the inputs. And with this, I want to thank you for your attention, and I want to thank my students who drove most of this research. It is Roxana Zerazi and Sahel Azizpour, my collaborators in Göttingen, Tatiana and Jan Lang, her postdoc, and experimental groups that provided us data and, true enough, the funding sources. Thank you. I see there is a question from Tatiana, if I'm allowed to moderate a little. Oh, wait, 1 second. It feels like I don't hear anything, or...
Yeah, okay.
Oh, no.
Go ahead, Tatiana, Adrian.
All right, so yeah, I want to ask real quick questions. So if you assume that monkey is optimal, then based on like how close in monkey data, I suppose you can estimate how close you are to the critical state. Would you be able to tell what would be the integration time window which monkey is using to make decisions?
Yes, 1 second. I share again we definitely have some idea about it. Oh, wait, I'll share the screen. Okay, just share this. Okay, so here we see the distance to the critical point of the all parameters that would be in a matching model for attend away and attend in. We see that in all cases, the best matched model moves closer to critical state, and it indicates actually a very fast processing there, such that there would be enough averaging. However, it might be that what we have as the real true noise in the input in this simple model that just encodes inputs is some kind of relevant computational information, so that it is not as detrimental to be that close to critical state for the even short observation windows, because the monkey doesn't have that much time to decide. So we would need actually a very fast processing for this distance to criticality to be actually optimal.
And I have two questions. First, I was so when you were defining what is a cortical brain state or cortical state, you were saying that it has to be low dimensional. Why do you think it should be low dimensional? This would be my first question, and what you can.
So I believe that this is basically our human bias. We would not be able to define what is the state. And definitely what we've seen, for example, in Tatiana's work, it's just 6 dimensions. This is as low as it gets for this huge amount of variability that we have in the data. So we have in the multiple sources indications that it's often sufficient to observe really low dimensional, if properly projected, low dimensional part of the data to understand a lot about the data itself, the decisions that animal does in the decision-making experiments and so on. On the other side, I believe we just, like what we want to describe by state is something that we can mark and label and understand. and this is the essence of what is happening. And what we see often is that essence is actually relatively simple or low dimensional than what we observe in all the noise. Sure enough, there is no like proof except of the set of now very extensive set of different observations where low dimensional representation after the proper transformation was already returning a lot of information about the system.
For me, this assumption can be a bit risky, because well, the neural systems are highly complex and have multiple parameters. Maybe we don't have access to them, and they can transit through different states, even having multiple parameters. They can change the parameters. Some parameters can be very important, some cannot, can be like unimportant. But we often try to understand the data with simple models that usually have two extremes, like as you said, like random or synchronized. And in between there is something complex, and because biological data is always complex, we surprisingly always fit the data close to the point of maximal complexity. So maybe this assumption that the states are low dimensional are a bit misleading, that we are using models that are too simple, that almost like most of the time we end up concluding that the system is at a critical point, for example, and it's maybe only due to the simplicity of our models.
I agree. There is some kind of our internal bias towards being happier with a simple explanation or being not able to come up with complex explanation. Though, say in a part of criticality, we don't really capture all of the data. We project it in a very simple space, and then we find it in one point in this simple space. This is not everything that is in there. Even if we describe the system by low dimensional representations, they are changing over time. Possibly also the important projection axis would change over time. If we can pin down something like a state, so I think it is a little bit about language, that if we pin down a state for some period of time, or maybe some part of the system and some period of time, we would most probably think about something that is describable in the low dimensional terms. But I'm happy to see the other examples, so see how to use the more complex descriptions at all.
I have just another question is, so like mixing Tatiana's talk and yours. So do you think that the drivers that Tatiana measured, the driver regions, they should have an autocorrelation that is much shorter than the other ones that are driven by recurring connectivity, no? This would be a hypothesis that you could test with Tatiana's analysis?
I'm not sure that is related this way. And in Tatiana's data, we have a very slow time scale that is defined by the rate of input of the data processing, like of the calcium indicator. So it is slow because we record it on a slow time scale, at least in some parts. I guess that if the recording would have been done on the individual spikes, this is a question to Tatiana and maybe for the discussion, then we would not have the time to get to the stationary state. So whether the driver parts should be faster, I think they should be fast enough to capture the timescales in the real world that are very different, and some of them are very fast, so we definitely need some very fast driving structures. I think some of them might be also slow. I'm not sure we can really match one to another that easily. Sorry, need to think about it. We can discuss it with Tatiana.
We have time for another question, Adrian, because Lana is asking.
Yeah.
I'm sorry. Okay.
Yeah, we can hear you.
I have a question related to your assumption that, not assumption, but basically your results showing, even in this slide, the difference in their response, more or less critical, depending whether the neurons are attending to that particular input that's being presented. So I would like to expand on that, considering the fact that usually in models of criticality and applied to neuroscience, we can usually see that the networks being analyzed are infinite or have properties that are not physiological or adaptable to experiments. So one thing would be, as you mentioned, infinite time to analyze the input that you have, but also having a infinite network of neurons that you wouldn't see the borders, and then you can analyze your critical process more accurately. So in that context, how do you think a network that, for example, would attend to different orientations in different regions by different cell assemblies of neurons, how would it organize itself to find critical states in specific locations, given that when we think of criticality, usually we think of this big network that would expand information in a very large space. But at the same time, you're now localized, that you have to process information, like different information in a very localized space. So how do you think these two, a little bit like opposing ideas could be integrated here?
Thank you, Fajin. Indeed, if we look at the world as a physicist, the critical point is defined only in the infinite system size limit. What people are using sometimes is the so-called Griffith phases, where or the investigation of how the same model, if we would phrase it as a small and match to the experiment, how would it behave if we would change it to the large system size and define the state of the small model by the behavior of the large model that it would have be if we would have an infinite network. On the other side, we could also have a critical process in a very simple, like single dynamical system that could be tuned close to bifurcation point. Each hair cell in the ear is considered to be at the critical point. So I don't see the necessity of having the infinite system to have a critical point. It's our inference of what's critical and what is not relies on the statistical methods that are, in most cases, well studied in the limit of the infinite system sizes. If we would look at the closeness to bifurcations, it would be possible very locally. This would be, another different way of defining what is a critical point, to be close to the bifurcation point. And this would also allow a big spread. Again, the model also, the modeling approach allows us to say, okay, if the local model would look this way everywhere, and we would have a huge model, this would be a model that is the criticality also statistically, if we use the same statistical methods. However, it is really hard to define. Here, we use a local measure, that is this autocorrelation timescale for our local recordings. And we evaluate how they behave locally. This is not the global state, and we also see that it is changing. The self-organization towards this state or the self-organization towards a change from this state to the attended state, we have this options that we found so far. Like basically there could be some top-down inputs, or there could be a neuromodulation. But we, like in the model, we could just change parameter. We don't know what it should be coupled to, and possibly we need harder models, including more components to capture the self-organization there if we want to have it completely internally driven. So if, I don't know whether I answered the question you asked.
No, I think it's close. I think ultimately it's more, it would be more related to the self-organization of the system, because you would need this, in my, the idea behind my question, you would need this switching between different states. And then I think that really relies on the self-organization, but thank you.
Thank you.
We continue the workshop with Enzo Tagliasucchi, who is a professor at the University of Buenos Aires in Argentina. And he will talk about how to perturb in silico to understand states of consciousness.
Great. Okay. Yeah, so you should be seeing my slides. Is that fine?
Yeah, perfect.
Okay, excellent. So I'm going to put a chronometer so I don't speak for more than what I should speak. Okay, excellent. So thank you very much, Adrian and Gabriela, for the invitation. I'm very pleased to present this talk at this symposium. So my talk will be about states of consciousness, but I will talk about human subjects. So my presentation will include at least some discussion of consciousness in terms of subjective reports. And then, of course, we will try to apply different models to human data as well, to try to use perturbations as at least as an interesting measure to disentangle different states of consciousness. So first, I would like to start with something that some of the speakers already discussed, but I believe that we should revisit perhaps when we speak about human subjects. The idea is sorry, I want to, oh, there it is. Yes, so the idea is that we want to define what is a state of consciousness, and we also want to measure different states of consciousness. And we already find difficulties because, as some of the speakers already mentioned, we really don't know what state of consciousness is or what the brain state is for that matter. So through time, we find different analogies, many of them coming from physics, trying to suggest methods to measure states of consciousness, or even to measure subjective states of consciousness. One of the leading analogies, I think, is the notion of temperature and the idea of measuring the temperature of consciousness. You'll find direct mention of the idea of building a thermometer for consciousness in the literature. This is quite common, actually. And this analogy is made in terms of temperature. So the idea is that there is a unidimensional scale that can be used to measure consciousness. So because of the reason, we speak of the level of consciousness in the same way that we speak of the temperature of water, for example. This is a single number that is supposed to capture what is the intensity of consciousness. Of course, different scales can be used. So you have the Fahrenheit scale, you have the Celsius scale when you speak about temperature. When you speak about consciousness, you will find different methods. For example, the perturbation complexity index, entropy-based measures, or even commercial measures such as the bispectral index, that using a single number, they pretend to capture, to measure, to be useful to measure consciousness. So as in the case of temperature, you have different scales. And of course, there is something that is implicit in this analogy, that is a reference state. So in the same way, you have 0 degrees for the Celsius scale, or you have minus 273 degrees for the Kelvin scale. Well, the idea is that you speak of increased or decreased consciousness in reference to a certain state. So of course, this if you consider what we just discussed in some of the previous talks, this is probably too simplistic because you know, if you walk through Germany in any large German city, you will find these kind of machines like a Wetterstation that they pretend to, or they actually claim to quantify the weather using 3 numbers, you know, temperature, humidity, and pressure. Or if you see the Olympics, you will see that you have different scores with different judges to quantify, you know, how good was a certain routine, or even personality in humans. You know that a single number won't capture human personality. So these are just examples to tell you that why is it expected that we can measure the weather using 3 numbers, but for something as complex as the brain or even consciousness, a single number will be enough. So this is probably too simplistic. So is one number enough? Probably no. So if you go and look deeper in the literature, you will see, okay, there are different other proposals, like using 2 numbers, the level of vigilance, you know, how responsive you are to the environment, and the level of awareness, so the intensity of conscious content. So you can place different well-known states of consciousness, like sleep, the vegetative state, in which you have vigilance, but at least we don't believe there are meaningful conscious contents, and so on and so forth. So you can try with 2 numbers. This, of course, also is limited. You know, why would it be possible perhaps to compare light sleep and REM sleep, or deep sleep and epileptic seizure? Maybe it is implicit in this kind of diagrams that you should be able to compare any 2 states, but perhaps there is partial order, so it doesn't even make sense to compare the intensity of consciousness between some pairs. And the whole idea that 2 dimensions are enough, it's also ad hoc. So of course, if 2 dimensions are not enough, you try with 3, and then you'll see other proposals, also ad hoc. For example, the proposal by Hobson of the AIM model, you know, introducing different variables that are related to different modulation by different neurotransmitters, and so on and so forth. So in the end, basically what seems more reasonable is to conclude that we shouldn't be thinking of consciousness in terms of a single number. And when I say consciousness, I'm speaking about states of consciousness that are extended in time, like sleep, anesthesia, and so on and so forth. So in a sense, when I say state of consciousness, I'm also referring to what the previous speakers called brain state, at least to some of those brain states. The ones that we see in humans, for example, that humans can distinguish in terms of their subjective experience as temporally extended states. So the idea is that a single number will be enough is probably too simplistic. So not long ago, Bain and colleagues actually proposed this idea, proposed that state of consciousness should be characterized by a number of dimensions, not a single one, and the profile of these different dimensions is what distinguishes between different states. So, you know, you will, if you plot deep sleep, you will have a shape here in this radar plot. If you plot, you know, a certain, you know, dreaming or psychedelic state or psychotic state in a patient with schizophrenia, you will have different shapes, and these shapes are what are distinguishing the different states. And this gets even further more complicated, again, when you consider a state of consciousness like some of those that I just mentioned, that are not reduced in terms, you know, they are not considered state of reduced or increased consciousness, like sleep or epilepsy or anesthesia, but they are different in terms of the subjective experience. So this happens, for example, for pharmacological interventions like psychedelics and other drugs. It's even more complicated to accommodate the idea of a single unidimensional scale to explain this very different state of consciousness. So the problem with this idea is that nobody really knows what are the dimensions, or for that matter, how many dimensions you need to invoke for this explanation. Actually, this was part of the discussion that Anna and Adrian just had. You know, this is really a low dimensional problem, or in the case that you actually admit that a few dimensions will suffice, well, what is the meaning of those dimensions? And for in the case of humans, it's even more complicated because you have to think of those dimensions, both in terms of something that will allow you to reconstruct from that low dimensionality representation the brain activity, but also you have to think of those dimensions as something that perhaps maps to subjective experience. So if we say, okay, 5 dimensions are enough to reproduce all these different states of consciousness, then for humans at least, for which we have very rich and detailed subjective reports of subjective experience, then the question arises, well, what are those 5 dimensions in terms of subjective experience? And really nobody knows this. I mean, this is not something for which we don't really have agreements. And it gets even more complicated, because in a sense, this is basically the problem of that we don't know what a state of consciousness is. It's easier to provide examples, to say, sleep, anesthesia, blah, 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 but it's much harder to give a nice explanation. I think Anna attempted to do that, and very nicely. It gets more complicated for humans, because it's not just a matter of, when you speak at least of state of consciousness, not a matter of explaining the brain activity from a reduced dimensionality representation, but also, again, coming up with a representation that makes sense in terms of subjective experience. And then you run into all sorts of different questions. For example, how long does it have to be a brain state to qualify as a state of consciousness? Like, is a deja vu or hitting your toe against a piece of furniture, is that a global modification of the content of consciousness? Probably yes. But does it last long enough? Well, maybe no or yes, depending on whom you ask. So we see that we find problems of granularity in the definition of state of consciousness. So I think that the problem behind all these, or at least part of the problem, is that we have only in part abandoned the temperature analogy. So when we say, let's drop this unidimensional scale, we are not really saying, it's not the same as saying, let's drop the temperature analogy, because we are still holding to a key component of the temperature analogy, that is the reference to an absolute state. And I think that this is the problematic thing, that the idea that there is something here in the middle of these diagrams, in the 0, and that all these dimensions we measure in reference to this 0 point. And of course, then finding which are the relevant dimensions is similar to finding, what is the center of this diagram. And this is complicated, and we don't know how to do it. So perhaps a better analogy is the idea of space for the metrology of consciousness. And you know that in physics, we can measure points in space from certain, with multiple dimensions, of course, 3 dimensions usually, but we also do that in terms of certain coordinate or center of coordinates. But we can change the coordinates, and of course, if we do so, the dimensions, the actual numbers, the coordinates needed to find these different points in each system of coordinates will change depending on where you are located. But there is something that doesn't really change. That is, of course, the distance between the points. This is what in physics we call an invariant. So the distance is the same. Doesn't matter where you're looking at the system. So the distance is an invariant, and it's of course independent of a reference state. The same happens if you change not the center of coordinates only, but also the system of coordinates. You can go from the classical orthogonal system of coordinates to more complicated, spherical, cylindrical, whatever you want to do, but the distance remains the same. So my point here is that perhaps there is a way to find these coordinates that nicely map to subjective experience, and that also you can understand in terms of brain activity. Perhaps there is a way, but we don't know yet. So we can postpone finding those dimensions if we choose to focus on the distance between states, because then we don't really need to find those dimensions. They're implicit whenever you compute the distance between states. So, and then of course, if you have a rich and detailed and precise and robust way of measuring distances, you also postpone the problem of actually saying what the state of consciousness is, because whenever you are equipped with a notion of distance, you can cluster things, and you can do hierarchical clustering, and then you can say that basically a state of consciousness is something that you can cluster at a given spatial and temporal grain, and then basically you choose how far in the dendrogram you want to go, and that is a practical operationalization of a state of consciousness that doesn't really require, at least in principle, finding which are those dimensions. So this is a way, at least the way in which we're going to move forward with our talk. So what follows in this talk is I want to show you some examples of this idea, this idea that if you choose to focus on distances between states, you can get useful information even though you don't really attack the problem of the dimensions. Then I want to tell you that certain different distances can give different information, and that computational models can be useful actually to introduce and compute certain distances between states, which are the perturbational distances. And then finally, I will go back again to the issue of the dimensionality reduction, and I will show some preliminary work trying to see if once you settle for a data-driven way of finding relevant dimensions to characterize states of consciousness, well, if in the case of human data, those dimensions actually make sense in terms of subjective experience and what we can say about them. So for the remaining of the talk, this is all the results I'm going to show have just been accepted for publication in PLOS Computational Biology. So you should see the paper online like in a week or so, I hope. So the data we studied in this paper was fMRI resting state data acquired in 3 different states of quotation mark reduced consciousness. And I'm using quotation marks because I just attacked the idea of a single unidimensional scale to measure consciousness, so it could be a little bit contradictory if I now use reduced consciousness. But again, I think you know what I mean. States, states like this. Sleep, for example, sleep has 3 different substages, and 1 and 2 and 3 of increasing depth. Sleep is reversible because you wake up, and it's unstable because you can wake up, by not very strong perturbations. At least if you're a healthy sleeper, someone clapping in your ear will probably wake you up. Then we have shear anesthesia. In this case, actually propofol induced anesthesia. We have 2 levels. This is data. This is data from my lab. This is data from the lab of Stephen Loris in Liège. So you have 2 levels of sedation, actually mild sedation and loss of consciousness. And this state is reversible because people, again, they wake up from anesthesia, but stable because, well, that's the reason why surgeons do not wake people, until they fall asleep to operate by the use of anesthesia because it's more stable. And then we have data also from Stephen and some from Jacobo Sid in Paris, of patients, brain-injured patients who have disorders of consciousness. So these patients are vigilant or can be vigilant, but there are no signs or few signs depending on the intensity. We have minimally conscious states and unresponsive wakefulness syndrome. These are 2 severities of disorders of consciousness, and depending on the severity, there are more or less signs of conscious content in the patients. So this is this can be unfortunately irreversible. So minimally conscious state might show some progress, but the unresponsive wakefulness syndrome is in general quite irreversible and is stable as well. So of course, from the point of view of subjective experience, we have the intuition. At least I have the intuition, and I think it's supported by subjective reports of larger populations of people, that at least some parts of these states of consciousness feel in the same way. Actually, they it's not that they feel in the same way, but they are in the same way in the sense that they lack subjective experience. So in terms, again, of subjective experience, they are similar. There is none or there is little. But in terms of brain activity, or in terms of the way, the route followed to actually get into those states, well, they are quite different. So it's an interesting question if whenever you are unconscious, there is only one way of being unconscious, or there are several. So this is, for example, a question we would like to ask. So if there are several, I mean, if there are different patterns of brain activity related to different states, it could be interesting to ask, for example, if these differences are related to some of these variables, like whether the states of consciousness are stable or not. I like to show this analogy from physics. So in physics, you under a gravitational field like the one in which we are all of us now, you can have different surfaces like this one here or this one here, and in these surfaces you can place two balls like this one here and this one here. And if you ask what is the level of energy of each of these systems, the ball placed here or here, the level of energy is the same. So you cannot distinguish between this system and this system just by looking at the potential energy of the balls. But of course, the system is not the same, because we know that the system is stable against perturbations. So if you kick this ball, it will tend to go back to this position. But if you kick this one, it will go away. So here is the conceptual idea I try to convey in the following slides. This is one of the reasons why it might be interesting to include perturbations when we discuss a state of consciousness, because again, the level will not be enough. Even if we choose to believe that level of consciousness is something interesting, important by itself, it will never be enough, because we also have to take care about the stability. So what we're going to do, our strategy to understand these different states, is to introduce different, at least 4 different metrics of distances between states and compare them to see if they are giving the same information redundant, or if they're given different information, and what is the meaning of that. So for example, one way to compare these states is something that we call correlation distance. And the idea is that you have fMRI data. So this fMRI time series you can map into a certain parcellation of the brain. And then this is quite standard. You can compute a matrix, which is the functional connectivity matrix, and subtract from this matrix the baseline. So if you have, for example, deep sleep, you have a matrix for deep sleep, and then you subtract the matrix for wakefulness. So you have a difference between the state of unconsciousness and the conscious baseline. And then what you do is that you have a certain pair of states. So you have the white columns of the resulting matrices for each pair of states, basically. So you take the one of the rows of these matrices, and then you compute the correlation between states of each row. You choose one row. You do this by rows because you want to map something in terms of regions in the brain, and there is one row in the matrix for region. And then you highlight regions with a high correlation. So the idea is that in the end, you can count the number of regions with high correlations and obtain a numerical index of the similarity between states. So for example, this is what happens when you do this for N1 sleep and N2 sleep. So each what I'm showing you here is the resulting regions for which the profile, the connectivity profile, meaning all the correlations of each region with the rest of the brain, is correlated above this threshold between these two states, right? So you have a certain extent of regions which are similar in terms of functional connectivity between these two stages of human sleep. If you go deeper, if you compare N1 to N3 sleep, which is a deeper stage of sleep, no wonder that you find less regions, because we know that N1 is farther away in terms of the depth of sleep, and by this there are many variables that you have to consider, but at least this is the convention in sleep medicine, that N1 is farther away from N3 sleep, so you see less regions here. When you compare these two states, they are closer in the progression of sleep. And then when you go and compare N2 and N3 sleep, basically you see that all the regions are highly correlated in terms of local functional connectivity profile. This is also reasonable because N1 and N3 sleep are very similar in terms of subjective experience. Basically, the N2 sleep is the state in which conscious experience starts to fade away. So then you can compare N2 sleep with loss of consciousness by anesthesia, and then you have less number of regions than when you compare it to sleep. And then you can compare deep sleep to loss of consciousness, and you have lots of regions because those two states are, again, the deepest in terms of anesthesia and sleep. And then you can compare the two different kinds of patients and see that they are very similar but very different to the other sets of states. So this is just a simple illustration that you can compare different states of consciousness simply by looking at the local connectivity profiles. This is empirical. There's no model here, no perturbations, just looking at the data. Other way to do this, to introduce a different measure of distance between states, is what we call classification distance. And this is that you train a machine learning model to distinguish each state of unconsciousness from its corresponding wakefulness. So you train a random forest classifier, in this case, based on this functional connectivity matrix, and use it to distinguish, for example, deep sleep, entry sleep from wakefulness. And then you ask whether the same classifier can be used to generalize to distinguish other states of consciousness. So you ask whether there is transfer learning between the classifiers. So, for example, this is the subtraction of the functional connectivity matrix for entry sleep minus wakefulness. You have here I'm showing the upper diagonal part because this is a symmetric matrix. Red and yellow is positive and blue is negative correlation, but this is again the subtraction of these two matrices. This is the same for loss of consciousness by anesthesia, subtracted by the wakefulness baseline. And you can see that there are some similarities already just by looking at the matrices. You know, you have this negative region here, this positive row. Of course, this is just by eyeballing the matrices, but you can see there are at least some structures similar. And then you can do the same for unresponsive wakefulness syndrome. These are the brain injured patients minus wakefulness, and you don't see this kind of structure. This gets a lot noisier. So when you introduce classifiers, what you find is something that actually reflects this similarity in the matrices. You can train machine learning classifiers to distinguish loss of consciousness from wakefulness, and then use them to distinguish deep sleep from wakefulness, and vice versa. But you cannot train a classifier to distinguish deep sleep from wakefulness and use it to distinguish unresponsive wakefulness syndrome from wakefulness, and vice versa. Same for anesthesia. So this is something that you could expect by looking at this. So this lets you introduce a metric of similarity between states of consciousness, which is basically the area under the ROC curve, so the IUC of these classifiers. And then we introduce a model-based distance, and this is when I think it becomes more interesting, which basically we first obtain a set of parameters that, using a whole brain model, allow us to reproduce the FC matrices of these different states, empirical matrices, and then compare how similar those underlying parameters in the model are. So the idea is that, of course, there could be lots of models we could use to try to reproduce the functional connectivity matrix. Much more complicated than this one. This is, I think, one of the simplest, or perhaps simplest model that captures a transition between noisy fixed point dynamics and oscillatory dynamics. This is a normal mode of Hopf bifurcation. Basically, you have a transition between fixed point dynamics and oscillatory dynamics at a frequency that is determined again by the empirical MRI recordings of each node. Whenever you move this parameter A, which is a bifurcation parameter. So if it's less than 0, you have stable dynamics, fixed, governed by noise at the fixed point. And if you have larger than 0 A, you start to see oscillations. So of course, each node has an equation that governs the dynamics of that node, but you introduce coupling between nodes based on DTI. So the idea is that you include empirical sources of information in the model to then be able to reproduce by optimization of these parameters the empirical as well FC matrix. Otherwise, you wouldn't be entitled to expect that your model can say anything in terms of realistic neuroimaging recordings. But since you include all these sources of empirical information, then you can expect that, at least to a certain extent. Of course, the dynamics are cartoonish, but they are among all the possible cartoons, the more realistic one, or the more suitable one if you want to capture this kind of dynamics. Then the optimization target is the FC matrix of this model, And then we don't allow each region to have its own bifurcation parameter because the parameter space will be huge, and this is a problem. But we allow different bifurcation parameters per within each one within a different set of anatomical priors, which are the resting state networks. So each one of these will contribute independently to the local bifurcation parameter. So we have an optimization problem, which basically we have a large set of coupled nonlinear differential equations. We have one parameter that relates to the scaling of the global coupling, and then we have 1, 2, 3, well, we have 6 possible parameters that contribute to the local bifurcation parameter of each node. So we have this 7 parameter space, and we use genetic algorithms and other tools to optimize and get quite reasonable reproductions of FC matrices from these dynamics. So once the models are fitted, we can then obtain the optimal model parameters and compare states simply by looking if the optimal parameters that reproduce 2 states are similar or not. So in the end, we have all these different ways of compute the distance between states. We have all the states in this matrix, and we have the correlation distance, the first one I mentioned, the classification distance, and the one I just mentioned, that's the model parameter distance. And what happens in the end is that if you do scatter plots of these matrix elements between all pairs of distances, you have, classification versus correlation, model parameter distance between correlations, and model parameter distance with this classification distance. So each point here is a pair of states, and you are basically asking whether knowing one of these distance matrices allows to predict the other ones. The answer is positive. So the correlations are not huge, but they are significant, and all these things are correlated. So the idea is that they are not really giving complementary information, or at least not giving complementary information above a certain threshold. So the information is similar between these matrices. So they are all correlated. According to all of them, the patients are separated, so they have a high distance, to all the other states. And then you see results that make sense in terms of neurobiology. That propofol is similar to intermediate sleep stages, or that propofol induced loss of consciousness is similar to deep sleep. So then we say, okay, we want to remember this slide in which we have these 2 balls, and we say that the level of consciousness is not enough, or the level of energy is not enough. We need to understand what happens in terms of perturbations. So what we do is we add a periodic forcing term to each pairs of nodes that are symmetric respect to the midline of the brain. So these are homotopic brain nodes. And we add a periodic forcing term, a cosine with a resonant at the frequency of the resonant frequency of the node. So each node has a resonant frequency, the one that was used actually to construct the Hoff model, the one at the frequency at which the node oscillates when the bifurcation parameter is positive. And then this periodic forcing has an amplitude, so it has a scalar term that multiplies the cosine, and we change that parameter across a wide range and see if at least for some reason, for some values of that parameter, we see that by including the perturbation, we can change the FC matrix so that now it's more similar to the FC matrix of the other states. So the idea is that we feed the model to a state, we introduce a perturbation, we explore, lots of perturbations, and we see for at least some value of the perturbation amplitude and for some choice of the nodes, we can modify the FC matrix that's reproduced by the model to look more similar to the matrix that corresponds to another different state. And in that case, we say, okay, we could induce a transition from one state to the other in the model. And then, of course, the change in goodness of fit with respect to the target state, the idea is how much we could make the FC matrix look like the target state is our idea of distance, basically, or similarity. If you can induce a transition between 2 states, then in terms of perturbation of similarity, they are close. Because even if their FC matrices are different, you can again transition between them by a suitable choice of simulated perturbation. So when we do this, basically we end up with a matrix between all pairs of states in which this is interesting. This is not symmetric like in the other distances, because here the target and the starting and the target end state matter, because it's the fact that you can induce a transition from, I don't know, waveforms to N1 doesn't really imply that you can do it the other way around. So it's not symmetric. So you can represent this as a directed graph. The directed lines tell you, okay, that you can induce transition from the starting node to the end node. So what happens here is that you see that wakefulness tends to receive most of the arrows, and that sleep, for example, tends to send lots of arrows to wakefulness. So the idea is that if you perturbate the model, you can transition from most states to wakefulness, although not for the patients. This is interesting. So at the end of the day, you can basically add another axis here called instability. And this idea of instability is basically how many arrows you get from a node to others. I mean, how easy it is to displace the model from that state to the others. And here in this case, you see that in terms of instability, N1 sleep, which is a transitional sleep stage, and this is quite reasonable, is the less stable, and the most stable are wakefulness and loss of consciousness, and also the responsive wakefulness syndrome patients. But of course, they are very different. So wakefulness is stable and conscious, and loss of consciousness state by propofol is stable, as we expected, but it has a low level of consciousness. And then finally, we can map the regions in the brain where consciousness is increasing or decreasing by these perturbations, and this makes sense in terms of, well, of what we know about the pathophysiology of some of these states. So, and the interesting point, just I'm close to the end, is that this perturbational way of defining distances between states is not really correlated. So these are the correlations I showed before for the other measures of distance between states. But for this new metric, these perturbational metrics, the correlations are close to zero. So it seems to give independent information. Just to finish up, I will take 1 minute or two to show some ongoing work. The idea is that if we go back to the idea of this low dimensional representation of brain states, well, we could, I wouldn't be too enthusiastic about the different dimensions that are introduced by neurologists or by cognitive scientists, because there's no guarantee that they will map into something meaningful in terms of brain activity or brain physiology. So I think that perhaps we should take a different approach and try to find low dimensional representation from the data itself. So if it's true that brain state is something that you should be able to reproduce from a low dimensional representation, of course losing information but not too much, then this is something you could approach. This is the paper where some of these ideas are based by some new and very interesting deep learning architectures like autoencoders, variational autoencoders, which are very efficient at reducing dimensionality of the data in a non-linear way. So, and they are also generative, so you can interpret the latent space, by moving these knobs and getting different results at the end layer, which are basically a combination of what is represented by each of the low dimensionality units in the middle layer. So what we show is basically that we can do this. We can encode the progression to deep sleep in latent space using autoencoders. So we go from deep sleep to wakefulness, and this is interesting because here you have two latent units. So even though we say that, okay, this is a representation of the progression from wakefulness to deep sleep, this is embedded in some kind of low dimensional algorithm. You can think there's a line moving here in latent space, and on that line you get an ordered progression of the sleep stages. So what we did, what we suppose is that, okay, if we feed the model and embed the results of the model in this latent space, then as we add a perturbation to the model, we will expect to see different trajectories in this low dimensional space, and these trajectories will encode as we increase the amplitude of the forcing term. These trajectories will basically represent what happens when you add a perturbation to the model fitted to a certain brain state. But since we are in low dimensional, two-dimensional latent space, we can really get some visual intuitions working for this kind of stimulations, because otherwise it's a very high dimensional problem. It's different to get an intuition. So this is the result of embedding all the states of consciousness that I just mentioned, from wakefulness to deep sleep, to the patient, unresponsive wakefulness syndrome. And you will see that you get an embedding that respects the level of consciousness. So you have wakefulness here, and as you move along this line here, you go towards states of deeper unconsciousness. And when you add perturbations to the model, I mean, you feed the model to these states, and you start increasing the forcing amplitude to up to asymptotic values, very large values, you will get trajectories. And what I'm showing here in these arrows is the mean ending point of the trajectory. So when you saturate the forcing amplitude, you get the last point in this trajectory as the forcing amplitude is increased. And what is interesting here is that all states, on average, moves to our wakefulness. But the problem is that some of these states are too far away from wakefulness. So even if you really kick them in terms of increasing the forcing amplitude a lot, you don't reach. Basically, you fall short. And this might be something you could think of a possible explanation of why these states are so complicated, so irreversible. It's not that you cannot displace the dynamics in the direction of wakefulness, but the problem is that you don't get there. They're too far away in this latent space. So this is an intuition of why some states of consciousness are reversible and some aren't. So, well, sum it up. I'm just, I think, over the time. So simply that unidimensional characterizations of the consciousness might not be sufficient. So we think of multidimensional characterizations, but the idea is that we don't know what are the relevant dimensions, so we can now think in terms of the distance between states. We show that this computational model allows us to introduce these perturbational distances between states, which provide complementary information to other kind of distances between states. And finally, this ongoing work suggests that perturbations actually displace all states of consciousness that were wakefulness, but sometimes this is not enough because the displacement does not really reach the cluster corresponding to wakefulness. So these are my colleagues and from the lab, my lab members and colleagues from different labs all over the world who contributed to this work, and thank you very much.
Okay, thank you, Enzo, for the presentation. I think we have time for one single question because we are running out of time. Tatiana wants to ask a question, I think.
Yeah, if I can. Yeah, very interesting. Thank you so much. So I was curious, like, how do you define what is the right perturbation? Because it seems maybe to go from one state to another, you need one type of forcing, and to go between two different other states, maybe it's also easy, but maybe you are just not using the right forcing. Like, how do you think, like, why there should be this universal forcing which you try to apply to any pair of states?
It's a very big question. The forcing, we didn't have much choice because of the model we decided to use. So this very simple model, including the perturbation, which is resonant with the frequency of the nodes, is probably the most reasonable. Otherwise, it will be difficult to get meaningful displacements of the models. But if you kick, as always, now when you have nonlinear oscillators which are oscillating a certain frequency, when you kick the node at that resonant frequency, you will get the maximum response. So if you change the frequency and you explore what happens as a function of the frequency, you will see that not a lot happens until you hit the sweet spot of the resonance frequency. So, of course, if we used more complicated models, we will have more freedom to choose and explore different stimulation, and of course, this is interesting, but maybe not so much again, because it could happen, and I believe this happens, that when other when ourselves also used more complicated models, sometimes you actually fitting the models and choosing the parameters so that reproduce the empirical data located at the bifurcation point. So if that's the case, all the complexity of the model doesn't really matter, because you can capture the same behavior with a normal mode of bifurcation. So I believe that could be the case, what's happening when people use more complicated models. So in that case, perhaps there is not so much that we have lost in terms of generality by using a simple model. But again, the complexity of the simulation depends on the complexity of the model, and in your case, that's the only reasonable option, I believe. Of course, there's another dimension in which it's true that perturbations could be different, and that's where you choose to simulate, which nodes you choose for simulation. And this is true. I mean, we only chose pairs of nodes, symmetric with respect to the midline, mostly because this is we wanted to choose a configuration that is not impossible to do in humans by non-invasive means. So if you could very easily induce this kind of stimulation using non-invasive transcranial stimulation. But if we also decided to explore, the exponentially large number of combinations of nodes for simulation, this, of course, it will be by itself a problem, because you have so many possible combinations of nodes, but also it wouldn't be so interesting because, again, in humans, you don't have the freedom to really stimulate whenever you like in the brain. So some of the results wouldn't be so interpretable in terms of simulation. So yeah, that's, I think, the answer. But of course, again, when I draw one of those arrows, or when I speak about the distance in terms of the model, it's true that it has to be interpreted within the context of a limited form of stimulation protocol that depends on the model, and also depends on the freedom you have to actually stimulate whatever you like in the brain, which for humans, that's not really possible.
I'm sorry, we have to move on for the sorry, Jacob. We have to move on for the talk of Valentina Dragoi.
All right, so I'm unmuted, right?
Yeah, can you try sharing the screen?
Yeah, I'm trying to do that. Let's see if this problem has been resolved from last time. Yeah, still have the same problem, but fortunately I opened to another system. So let me see if this one's available. Just give me a second. Sorry for the delay.
Did you try the shortcut? Ah, yeah.
You see it now?
Yeah, super.
Super.
Yeah.
Let's see if I can. All right. Can you hear me?
Yes, perfect.
Again, this is like, let me see if I can use a mouse. Can you see the mouse moving on the screen or not yet?
We can see the
mouse, no?
Mouse, no.
Mm.
Yeah, no, we see it. Now we see it. Now it's okay. Yeah.
Fantastic. All right. Thanks, the organizers, for making this possible. And I believe last time when I spoke here was also in Paris, and I hope next time will be in person. Okay, so for those of you who don't know what my lab is doing, we're interested in studying the relationship between information coding across circuits in the cerebral cortex and behavioral decisions. And we're employing a variety of techniques to understand, the more and more granular resolution as we move along and technology progresses from, in this case, from primary visual cortex to frontal areas, both feed-forward and feedback projections, including the specific manipulations, causal manipulations to understand how circuits generate behavior and how behavior changes circuits. So The reason I'm showing you this slide, this is a recording of 24 cells in primary visual cortex of awake monkey. The monkey is fixating for a period of 10 seconds. And the point I'd like to make here is that the state of cortical networks could change randomly during the time course of fixation. In this case, for instance, in the red box, you see that in state 1, neurons tend to be primarily synchronized, whereas in state 2, represented by the blue box, neurons tend to be primarily desynchronized. So it is conceivable that depending on the state of the network before stimulus presentation arrives, stimuli could be coded differently, right? And then possibly this could influence behavioral decisions of the animal. So my lab has been interested for a long time in state dependency, and for a number of years we look at state dependency in the context of adaptation. So how adaptation changes responses, correlations, and coding accuracy. But there are more dramatic changes in the state of cortical networks, and this most dramatic example, this is from a review paper by Harris and Thiele. For instance, in the top panel, the mouse is during sleep, and networks act in a synchronized way, whereas cortical networks are desynchronized, primarily desynchronized during wakefulness. Okay, so to outline my talk, in the first part, I'll refer to published work in my lab discussing synchrony in visual cortical populations during wakefulness. And in the second part, I'll refer to work in progress and some published in which we're discussing this issue of synchrony and the cortex in free moving animals. So we look at how technology will evolve from head fixed, restrained animals to freely moving unleashed animals. All right, so the first point I'd like to make is that there are different timescales when we refer to population synchrony. So again, I'm going back to the example I showed you early on, and I like to distinguish between examining population synchrony at the longer time scales, hundreds of milliseconds to seconds, as shown here in this blue box, and then I'll refer to the much shorter time scales at the millisecond scale, between 5 and 10 milliseconds. All right, so the point I like to make, just to preview, is that the good is, or the beneficial effect of synchrony reflects at the shorter time scales, in which we are discussing in the context of spike timing synchrony that is beneficial for neural function and behavior, whereas at the longer time scales, in which we're essentially capturing fluctuations in population activity, this is essentially detrimental for function during wakefulness. Of course, this is important during sleep, etc. We'll show later, and as Ansel's talk beautifully highlighted. All right, so this is Charles Beeman, has been an MD PhD in my lab, MD PhD student in my lab, and his brain sometimes goes from different states of wakefulness, highly aroused, large pupils, large eyes, to sometimes he gets drowsy. So he wanted to analyze this, the states of wakefulness in the lab, and he designed this experiment in area V4 of the monkeys. So he inserted multiple electrodes in V4 while employing a delay measure sample paradigm, so in which Charles had different patches of natural scene presented on a daily basis. So they're presented for a fraction of a second, shown here as target and test. And then in between, there was a delay period of about 1 second. It's variable between 800 and 1250 milliseconds. And what Charles did was to essentially look at the so-called delay activity between the target and the test, okay? And then what he observed is that there is a large variability in the population activity during this delay activity. So sometimes ranging from desynchronized activity, here captured both in terms of LFP and spiking activity. And by the way, this is an example in one trial recorded from 13 cells, right? And of course, same session. And as shown here in this panel, sometimes in a different trial, in this case, trial 217, population activity is highly synchronized, which is essentially reminiscent of more sleep states. Okay, and then on the bottom here, you see the average population activity from all these 13 neurons, and you can see clearly the distinction between desynchronized and synchronized activity that is reflected into just visually looks quite strikingly. right? And Charles has developed metrics to capture or to measure the degree of synchrony in this in the population activity by defining the so-called population synchrony index. And this is described all in this paper. I won't go into details, but I mean, there are many ways to capture that. For instance, take the average population activity, do Fourier analysis, and so on and so forth. So, and they all converge. That's a good thing. And for instance, you can see here in the so-called desynchronized trial, this is characterized by a population synchrony index of 1.19, which is pretty low. Whereas in a more synchronized trial, this more dramatic example, we see that the PSI goes at much higher levels. What I'm showing here is a trial-by-trial changes in population synchrony index here on the top panel, where all the trials are actually presented. Okay, so and we further distinguish between desynchronized and synchronized trials by analyze them separately by essentially taking everything below the median as being desynchronized and above the median as synchronized. And as you can see in this panel, there's a significant difference between synchronized and desynchronized trials within the same session, but these values are very different from those that one can record during the resting state. So this is monkey in a chair simply falling asleep, and then values are much higher simply, as expected, of course. All right, so our first intuition was that these more synchronized states may be related to maybe states of diminished arousal of the animal, maybe it's falling asleep or so. And then we performed a pupil diameter analysis as a function of PSI within the same session. Just to remind you, pupil diameter has been correlated with states of arousal, large pupil meaning high arousal. However, we failed to find a significant difference. I also confirmed this using EEG recordings on multiple sites, and then we essentially concluded from this analysis that what we're capturing here would be this changes on trial-by-trial basis in the state of cortical populations that may not necessarily be related to the changes in global states due to arousal, which I'll discuss later. So yeah. Anyway, so yeah, we further analyzed, Charles further analyzed the synchronized and desynchronized trials, and he found that populations of neurons extract more information from stimuli when local population and before is desynchronized. And as you can see here in the top left panel, I see this is the firing rate in the delay period in desynchronized versus synchronized trials, and you see that most points are below diagonal, indicating that, of course, firing rates are higher in the synchronized trials as expected, and also noise correlations are lower in the synchronized trials, so there's a statistically significant difference. And altogether, responses and correlations will sum up and allowed us to measure decoder performance, so the accuracy to which the population of neurons can distinguish between successive stimuli. And you found that in the desynchronized trials, the performance of the decoder is significantly elevated. And then the next step was to look at the behavior of the animal. Is the animal able to be the decision of the animal, the probability of a correct response is going to be influenced depending on whether the trial is, or the population activity is synchronized or desynchronized. And we found that, and the answer was yes. So as you can see in these top two panels, both percentage correct responses and behavioral thresholds were such that behavior was improved in the desynchronized trials. Again, I would say expected, but at that time, 2017, we did not have evidence that there was a direct link between the population activity and behavioral performance in terms of trial-by-trial fluctuations in population synchrony, when synchrony is measured at the longer time scales. So, and then we have some other analysis in the bottom panel described in the paper. I won't describe them here, but those interested can look at the paper, and they kind of discuss the robustness of these results. Okay, so the point I want to make with this, or the point I made with this previous few slides, is that in synchronized cortical state, we have those conditions that are essentially detrimental for behavior. And again, this is when synchrony is measured at longer time scales, with hundreds of milliseconds per second. In the next few minutes, I'll refer to much shorter time scales and make the point that this is, if we're going to measure, to look at synchrony or spike timing synchrony at this time scale, things are going to be much better in the sense that this is going to be beneficial for neural function and the behavioral performance of the animal. Right, so the question is, what is the significance of spike timing? We call it coordination, right? This is an example from one of the Pascal's papers in which we have essentially three populations of neurons, A, B, and C, colored differently. So A and C are synchronized, as you can see in this figure, whereas B and C are not synchronized in their spike time. In fact, they're anti-synchronized, right? Out of phase. And as you know, we all know from the field, from decades of research, good things are happening when neurons are synchronized, when populations of cells are highly synchronized in spike timing, and not so good things are happening when they're out of sync, right? So one major idea that has essentially emerged in the field over the past few decades is that synchrony is beneficial for behavior. However, we scratched the surface of this idea and realized that in the published literature, most of the evidence is actually coming from LFP type synchronization or coherence, or spike LFP coherence, but not so much from spike-spike synchrony or coherence. Or in those studies where there is a change, the changes were extremely small. So we were wondering why. And we realized that one of the main limitations of previous studies was the exclusive focus on pairized spiking events, right? But we know that, you know, it's just to look at population of cells. In this case, it's this cartoon example. One can identify pairs as well as higher order ensembles, such as triplets, quartets, and higher orders. And this essentially begs the question of, you know, if you were to measure and quantify this higher order event synchrony at the millisecond time scale, can you see any impact on the behavioral performance of the animal? Or not. So Neda Shahidi, with the help of Ariana Andre and Surin Pojoga in the lab, have done this dual recordings in areas V1 and V4, pretty much a task similar to that described previously, in which a target and test were briefly presented for a fraction of a second, and then there was a delay period in this case, and we had lots of trials, so animal was able to make errors and correct responses, obviously. And And what was distinct in the study was to essentially focus on or to distinguish between pairwise events and higher order coordinated events. So what I mean by coordinated event here is that regardless of regardless of the identity of the neuron, okay, we simply counted how many, say, triplets, quartets, higher order events were detected significantly during the time course of the experiment in different time epochs. And the main question was that, you know, is there any information that is encoded in the coordination rates, or is that just simply an epiphenomenon of neural spiking, right? Because we know that when stimulus is presented, neurons will fire at a higher rate, and of course, the coordination will be higher in those epochs. And I'll show you a slide that supports this point I just made. So anyway, we first confirm the results of previous studies, including ours, that stimulus identity is encoded in the population mean firing rates, right, which is expected. So here we again distinguish between correct and incorrect trials in, of course, these two areas. So we found that decoding the mean population activity on session-by-session basis in V1 and V4 yields essentially significant decoder performance, which increases with time, both in V1 and V4 for correct trials. However, while V1 is still able to extract stimulus information from in the incorrect trials, V4 fails to do that. And we reasoned that this is so because could be several reasons, but the main suspect for us was the extent of coordination or information transmission between V1 and V4 that we reason that is faulty in incorrect trials, right? Like no coordination or improper coordination which could either express between V1 and V4 or within V4 itself. So we further examined this question, and we first asked whether the spike time coordination or coordination rates will encode stimulus identity, which we previously found that is encoded in mean firing rates of population activity. And this is what a point I made earlier, which is when stimulus is presented, there is a high coordination rate as expected, so neurons fire in synchrony. and then the amount of synchrony will decrease. Okay, and that's why we had our measure as normalized coordination rate, just to account for changes across sessions, etc., and to be able to average results across sessions. So we failed to find that spike-time coordination is or can encode stimulus identity. So we cannot distinguish between stimulus 1 and 2 just observing the number of coordination rates, regardless of whether the events were pairwise or higher order, right? It's shown in this plot on the right. Right, so between stimuli 1 and 2, there's no statistically significant difference, regardless of the order of the event. However, we did find changes related to perceptual accuracy. Let me explain this somewhat busy slide. So this is, again, dual recording, same session, V1 and V4, 9 cells in V1 and 12 cells in V4, with other recordings with larger ensemble sizes. And we distinguish between correct and incorrect responses and colored the second order coordinated, pairwise coordinated events in blue and higher order. So here we lumped third, fourth, fifth, etc., which later kind of broke down into different orders. So we lumped them as higher order coordinated events. And like I said, we did this separately for correct and incorrect trials. And visually, if you look at V1, there's not much to see. It's nothing remarkable. So regardless of whether we focus on pairwise or higher order events, we don't see a difference between correct and incorrect trials. Whereas in things in V4 look a bit different in the sense that there is maybe the same degree of blue in both correct and incorrect responses, but there's more red in correct compared to incorrect trials. So in other words, you find in this example at least, in this session, you find higher rate of higher order coordinated events in correct trials compared to incorrect trials. And we further confirmed this for a number of sessions that we recorded for our in 2 monkeys. And we essentially look at the normalized coordination rate, correct minus incorrect responses. And we focused on the test stimulus, which is right before the animal makes the decision. And we found that in V1, there's no difference between correct and incorrect trials in terms of second order or higher order coordinated events. But the difference was significant in V4, but only for the higher order coordinated events, not for the second order events. And of course, we did a number of controls to make sure this is a real result we can trust, and we did also some modeling work. It's all described in the paper. The good thing is that this happens in the absence of firing rate modulation. Okay, so while average firing rates cannot distinguish in in our sessions between correct and incorrect responses, we did find a significant difference in spike timing synchrony, but only for higher order events and only in area V4, not in area V1. So we further looked at the coordinated events between areas, so between V1 and V4, right? So again, these are recording that is performed at the same time. Again, going back to this cartoon in which we essentially shifted it to spike trains in V1 and V4 by variable time lag, tau. And so when tau is positive, this means that V1 fires first and V4 is second. So V1 leads V4. And when time lag is negative, V4 fires first and V1 follows, okay, as expected. And the results are shown here on the right, okay. And we represented as a function of time lag and actual time from test stimulus presentation. The Z was the coordination rate, correct minus incorrect. So whenever we saw a change, we plotted here, we averaged across sessions. And the remarkable result in our mind was that we found a significant difference between coordination rates between correct and incorrect trials only in the feed-forward directions for positive taus, but not for negative taus, not for the feedback communication between areas. And to us, this was somewhat surprising because if anything, we expected that, if you want to look at the change or influence on behavior, what's likely to mediate behavior is feedback projections, right? So let's say attentional effects or behavioral context effects are coming through feedback projections, but that's not what we found here. We found that the feed-forward communication, which is in fact, extremely important to carry information in a rapid manner, but stimulus is most sensitive to the spike timing coordination. This is not to say that feedback is not important, it's just that spike timing coordination is not carried by the feedback, at least with respect to the data set that we generated here. All right, so just to conclude the first part, first point I want to make is that the desynchronized cortical state is associated with increased firing rates and decreased correlated variability, and altogether they create conditions that elevate network and behavioral performance. And the second point I want to make is that we found here, at least with respect to visual cortex, differential functional roles of spike counts and spike timing. And this has been a very, I would say, deep question, has been general theme, you know, is it more information on spike counts or spike timing, which one is prevalent? So we found that both are important. That is, spike counts carry information about stimulus identity, whereas precise spiking coordination within cortical populations carry information about perceptual accuracy. And we discovered later that, in fact, a similar conclusion has been reached with respect to motor cortex, where the similar strategies being employed by motor cortical nerves. So the next challenge for us was to, of course, there are many challenges with this, just continuing this type of analysis and experiments, but if you want to take it to another level, is to examine this relationship between population synchrony and the decision-making and stimulus encoding in freely moving animals. And this is the second part of my talk. Okay, so here we have two approaches in the lab about the relationship between synchronous fluctuations of population activity and behavioral performance and neural coding. And one is Russell Milton, a PhD student in my lab, has almost single-handedly developed this monkey treadmill setup in which animal has head fixed, which is clear advantages because we can present precisely controlled stimuli at millisecond resolution, while the animal is simply moving on a treadmill. We can vary the speed of a treadmill. Animal can control it or not control it depending on the nature of the experiment. But the good thing is that right now it's a quite reliable setup that allows us to measure locomotion, right? Here on the left, we're showing the kind of our goal here is to record from, to implant multiple chronic UTA arrays in multiple areas. Right now, we're only able to do it in area V4 and dorsolateral prefrontal cortex, but also plans to record in V1 and posterior and temporal cortex. So anyway, so just to describe what is shown here, the point I want to make here is that head-fixed bipedal locomotion modulates arousal and firing rate. So we were able, using the pupil size, right, to pupil diameter, to measure or to find that walking epochs here represented in red are correlated with a large pupil, but which indicates that it's a state of high arousal, as expected from rodent experiments, right? At the same time, population firing rates, this is mean population activity from tens of channels. These are, by the way, experiments in area V4, are elevated, while population synchrony index, which again, I want to remind you, represents a measure of synchrony or lack of synchrony, is reduced during walking, which confirms that in the so-called high arousal states induced by locomotion, we find firing rates and high degree of desynchronization in cortical populations. Our second approach is to fully unrestrained animals, and to perform wireless recording of population activity. And this is, let's see, again. This animal recorded what you're hearing, spiking activity in dorsolateral prefrontal cortex, or while animal is freely moving between two response boxes, box 1 and box 2, in a foraging task, and we have, we have a variety of experiments along these lines that range from natural viewing, foraging, social cognition, and sleep, right? So Russell Milton and Neda Shahidi in my lab, Neda now is in Göttingen in Germany, they've developed this, they put together the system that allows us to take advantage of wireless technology to have chronic UTA arrays. In this example, implanted in the dorsolateral prefrontal cortex. And like I said, we have examples in the lab now in area V4, simultaneous recordings, but we plan to go to more such arrays implanted in many areas. And the wireless transmitter can transmit spiking and LFP information while the animal is moving in the cage. We have cages of different sizes, like small cage and large cage, depending on the purpose of the experiment. While wireless eye tracker is measuring events with high temporal resolution, not high enough for our liking, but still enough to detect eye movements, measure pupil size, etc. And an overhead camera is able to measure the animal movement. So we have this fully integrated system that all of these components interact in real time that allows us essentially to know what the animal is doing on a, I wouldn't say millisecond, but maybe every 10 milliseconds or so, we know what the animal is doing. And therefore, this can allow us to distinguish between different states of wakefulness, which I'll mention a bit later. Just to show you an example here for 10-second recording in DLPFC during wakefulness and rest. An animal is, lights are turned off, animal is sleeping spontaneously. And in the desynchronized cortical state, firing rates are elevated, and you can just visually look at this, these plots. And synchronized states clearly see synchronized activity during rest, as expected, but lower firing rates. And like I said, we're able to look at essentially or to employ frame-by-frame analysis to look at the difference in pixels and have a threshold, distinguish between active and quiet wakefulness, and distinguish those with rest. And as you can see here in prefrontal cortex, we find that firing rates are elevated in active wakefulness, somewhat reduced in quiet wakefulness, and much more reduced in during sleep. This is recording of several hours in our setup, and we have many sessions of such recordings. And simultaneously, population synchrony index is reduced during active wakefulness and elevated during sleep. And this is significant across sessions. This is 2 monkeys, etc. All right, so we also decoded in the same style as Enzo's nicely showed in his presentation, essentially the behavioral state of the animal. We're able to decode it using just simply observing spiking activity and LFP. And in both cases, here the chance level was essentially 33%, or only 3 states: quiet wakefulness, active wakefulness, and sleep. And spiking activity, probably not unsurprisingly, would perform better than the LFP. The LFP is in decoding between the different states of the animal. All right, and we're currently using Deep LabCat to identify the targets of visual fixations and finally help answer this long-standing question of how do neurons in multiple cortical areas encode visual stimuli as the animal is interacting with the environment. And as most of you know, natural vision has been primarily studied in pretty much still lab conditions, so natural scenes presented on a computer screen, but there's been a dream of neuroscientists to look at this question in 3D objects in real life. And I hope we're getting closer to answer this question. Just to conclude the second part, we're advancing now towards a high-yield, fully wireless recording approach that will integrate motor, ocular, neural, and behavioral events in the second time resolution. Current success is to record from 2 areas. We plan to go to at least 4 areas and increase the density of the recordings. And one limitation is also we can record now on surface, but we like to go volumetrically. And of course, future challenges is to be able to also causally manipulate circuits in real time using wireless stimulation to activate and inactivate circuits, either electrical or optogenetically. I think we're not that far to do this optogenetically. And finally, to yield cell type specificity. All right, I'm not sure how I'm doing the time. Yeah, not bad. So anyway, I'd like to thank my lab members. Collectively have allowed us to perform this work, and particularly the people highlighted here in red. Melissa French, PhD student in the lab, she's involved, and Russell Milton have been currently involved in wireless technology and free moving experiments. Melissa is doing currently social cognition experiments. Russell is continuing with the treadmill experiments. Ariana Andres in Projoga with the dual recordings and optogenetics that I didn't have a chance to discuss today. And Neda, of course, great thanks to Neda. She's at the University of Göttingen in Germany. She's been instrumental in developing the wireless technology in the lab and, of course, the various funding sources. And thank you for your time and attention. I'm ready for your questions. Thank you.
Thank you very much. Perfect timing. It was perfectly timed. So if anyone has questions, I have one and I don't know, have emoticon to raise my hand, so I'm queuing, but if anyone wants to ask first.
I see Tatiana had raised her hand, but Gabriela, do you have a question?
Tatiana, yeah, go Tatiana, and yeah.
Oh yeah, thank you for the very nice talk. I was curious, so in the first part of your talk, you said that synchronized state on one hand is detrimental to encoding. I was curious, is short-term coordination of spiking activity, I forgot how you exactly named it, but did you ever look at whether existence of these highly synchronous spike events is higher during synchronized or desynchronized states? Because my intuition would be that during synchronized states, for brief periods of time, the neuron would conspire to fire all together, and then you get this like highly synchronized spiking events. But maybe overall, if you look in encoding through the entire longer period, the encoding would be worse. So I was just curious whether this short-term spike synchrony is more likely to happen during synchronized or during desynchronized state.
Yeah. So this is a great question. In fact, I knew you were going to ask this kind of question, knowing your work, and of course, we had many discussions along the subject. So when I talk about synchronized or synchronized fluctuations in population activity, I'm defining this at hundreds of milliseconds, right? Whereas in your work, I think you're somewhat shorter. So I think your time course is in between millisecond, which is in our case 5 to 10 milliseconds, and like hundreds of seconds, right? So you're more like in your case, like what, 140 milliseconds or so, and your synchronized states are somewhat different flavor than those that we're recording, right? You're doing this with laminar probes. But anyway, just to directly answer your question, we find this highly synchronized short-term events to be more prevalent during desynchronized cortical states, right? And I think in the field there is this sort of language mischaracterization when people talk about synchrony. No one knows what we're talking about, right? So in my mind, we should differentiate between synchronous fluctuations in population activity, which are occurring at hundreds of milliseconds to seconds, or even minutes sometimes, versus highly synchronized short-term events, like at the millisecond time scale. So those events that occur at the millisecond event do occur in the desynchronized state, while measured at longer time scales, not in the synchronized states. Not to say that they're absent, but they're more prevalent. And we're now looking at the differences, and hopefully we'll be able to publish the results. But yeah, great question.
I guess my question will be very similar to Tatiana about the time scale. So first of all, many thanks for the talk, and I'm super fan of the work. But I wanted to ask, so in this 2017 paper, there is a wake state when you have trial by trial fluctuation in this brain state, and it's desynchronized or synchronized. And then this like the newest paper from 2020, when you record this long-term fluctuation associated with global arousal in freely moving animals. You have this state of wakefulness, which you divide for quiet and active and rest. And I wanted to ask you how you combine these two papers. Like, I mean, this synchronized paper from 2017, it's like very local and it's very short time scale compared to the newest finding. So my question is like, how do you see it? Like, for example, this active wakefulness, it's the time when we have these desynchronized, synchronized switches on the trial by trial basis, but we don't have it, for example, in a quiet wakefulness and in the rest?
No, it's a great question again. So again, it's about timescales, right? So in the 2017 paper, which is one that I'm showing here, Biman et al., the animal was highly motivated, received high reward throughout the trials. The animal basically had no changes, no reasons to change arousal level, right? Was as motivated as it could be. And we did not see any changes, significant changes in reaction times or other measures that would be indicative, or eye movement quality, fixation quality, and pupil size. Like I said, were not statistically different across trials. Now, having said that, there are this writes on top of this kind of longer time scale changes in arousal induced by, one example is locomotion, but could be other reasons like reward. We know reward is a very important variable that controls arousal. Could also be behavioral state, whatever that is. So animal gets aroused, sexually aroused, or something that it likes or expects something that will happen. So I think they can all coexist, right? In other words, in states of high arousal, we have this micro fluctuation in the state of cortical populations, which are generated by and we know that they exist, this fluctuation in membrane potential of individual neurons that we know that are synchronized. And they've been characterized in the past by so many studies, right? We know that membrane potential is, and spike activity neurons is not flat. I mean, there are sometimes large variations, even when the state of the animal is strictly controlled for. Okay, so this is what I'm talking about in this study, 2017 study. Whereas in 2020, we're essentially identifying much larger changes in the state of cortical networks as induced by locomotion. Okay, and active processes such as animal doing something active like foraging or interacting with a conspecific, versus quiet wakefulness, in which arousal is much lower. It's not to say it does not exist. But on top of all of this, we have this kind of this riding wave, this changes at the memory potential level, essentially, right? It could generate changes in spike activity of cells, and these changes are synchronized. So I think it's a challenge for computational neuroscientists to be able to model these events that occur at this time scale, in which essentially there is endogenous waves of synchrony, right? So. And then they could coexist with changes that occur at larger time scale that are induced by arousal or activity, or some animals engage in something of interest or not, or attending. So I think the field is now is beginning to understand the interaction between these different cortical states that are that occur endogenously, that are due to intrinsic fluctuations in the spike activity of individual cells that are correlated, and more global states that are induced by arousal, wakefulness, etc., sleep. Yeah, this is fascinating. I mean, I think we're just scratching the surface now, but I think a lot of work is
Yeah,
done,
I think so.
including for multiple areas. Sorry?
I have 1,000 more questions, and I think
Sure,
like,
yeah.
I think it's a very good time to start the general discussion now. Adrian, do you agree?
Yeah, sure.
No, because it looks like.
Yeah, I think I agree with this. It's a good idea. So I guess we can unshare screen.
The reason why I ask, someone else wants to ask. Any questions from the public? The reason I ask about this, like, how do you place this relatively fast synchronized switches between brain states and this free moving animals study? It's because, there are also like recently studies showing that even if the animal performs the task, like, during one session, we see the slow fluctuation in like I'm referring here to the slow drift paper from Matthew Smith laboratory from Pittsburgh, that that we have this internal state, even within one session lasting 2 hours, when this brain state impacts our behavior and the population activity of the cortex. And given this, we cannot say that it's like resting or quiet wakefulness. It's like wakefulness, alert wakefulness, but nevertheless, we see this internal brain state changes. Would you call it also like, do you see something like this during active wakefulness, that you also have, on top of this, you have low fluctuation on the population activity in this state?
Right. So what Matt is observing, I believe you're talking about his neuron paper recording in V4 and DLPFC, right? And
Precisely, yeah.
that. So sometimes neurons at the population level, but it's not clear, but individual cells show drift in firing, right? If you're recording for multiple hours, usually it's not after 1 hour or within the 2-hour frame, but it's mainly observed for long-term recordings, 4 hours, 3 hours or more. So you see this gradual increase in population firing rate, either going up or going down. That's why in many studies we use the so-called, people are using a so-called detrending. So to remove this long-term fluctuation. So let's say if you're focusing on correlative variability, you don't want to be influenced, the results to be influenced by this co-fluctuation, the slow fluctuation and the firing of cells. You want to remove that. But yeah, so answering your question, yeah, we do see that when we are focusing though on, how to say, on the relationship between neurocoding and behavior, we're trying not to, or to eliminate those type of sessions, right? Because we think that that would contaminate the results. And this is typical in electrophysiological studies, right? You don't want those kind of data to contaminate the results because your conclusions could be driven from just observing an effect or phenomena that modulates the entire recording, right? So there's a slow drift in population activity, either up or down. We want stable to be, we like firing to be stable across trials. So again, two things. One, in medium-term recording sessions, we do not see this drifting. Occasionally, you do see it, and you are trying to, number two, trying to remove those recordings because they may contaminate your results. But if the focus is on specifically those states, of course, you don't want to remove them and you want to study them. So I think we should study them and to see how we can, how those slow fluctuations could coexist with whatever we're measuring, how they influence what we're measuring. And I don't think that's study has been done to reconcile this to, I wouldn't say reconcile, but essentially to see how this slow fluctuation could influence the results derived from very precise, say, spike time synchrony. Yeah.
Thank you. Any questions on the audience?
I have a question to start the general discussion. I have a question it relates to Tatiana's work and Enzo presentation. So that Tatiana Engel showed that there are in the model that she derived, there are drivers and brain regions that are driven by these drivers. And so a question would be like, considering that the work of Enzo has shown that perturbation is a good way to distinguish between brain states. Maybe a hypothesis to test would be how is the response of the different regions in the network studied by Tatiana, and whether the drivers are those that are responsible for a phase transition that you can maybe quantify using complexity measures or whatever preferred measure you have to distinguish the states. So do you think that this could be a good new like future investigation?
I will briefly answer. So in our model, what we tried to do, of course, was to see if the likelihood that a perturbation induced a transition between states was related to some topological feature of the structural connectivity network. We looked at the most basic features like degree, centrality measures, and things like that, and we did not find any obvious correlation between that and the likelihood of inducing transitions. But this was very, very briefly done. We did not go in depth. So probably yes. I mean, there has to be some structural feature of relevance that tells you, I believe, tells you whether the stimulation, the external stimulation, will be amplified or not, I think. Yes.
I would also like to answer that definitely it's a very strong model prediction, right? That if you perturb a particular area, which would be predicted by our analysis, you will see effects not only locally, but you will see a distributed change in neural activity all over the cortex. And what we would predict that stimulating driver area, a smaller perturbation should be able to elicit a larger effect compared to if you would be perturbing a driven area. So we are very interested to test those predictions in collaboration with experimentalists, because now you can not only do wide field imaging recordings, but you can also do optogenetic stimulation in specific local regions in the cortex and see what effect it will produce all over the cortical surface. With respect to human data, I guess there we are a little bit limited by the quality of anatomical connectivity data which we have, because we have to rely on DTI imaging connectivity. And the major limitation of it, in my point of view, is that it is symmetric. So our analysis shows that particular set of connections in the cortex, which is clearly non-symmetric when we look in the cellular level connectivity measured with tracer injections, is important for predicting activity, right? So like neither of our results would be possible to observe if we would just symmetrize the connectivity matrix. So I guess if we would have access to better measurements of connectivity in humans, then we would be able to kind of get more precise relationship between activity and connectivity. But with respect to brain transition, I guess now we do realize that they do not occur simultaneously all over the cortex, that there are some regions of the cortex which are more likely to support specific states, such as sleep and wakefulness. And clearly there is a tendency of those states to be initiated in specific region and then propagate. We are very interested in this question, but I guess as a field, we just in the beginning of understanding the spatiotemporal dynamics of state transitions. And I guess this is what Enzo is capturing, these changes in functional connectivity in these different states. So it's a very interesting approach.
So if I can say something along the same lines, we've started a couple of years ago to apply this perturbation techniques to essentially read out the state of cortical networks. And we have a paper now, the second round of review, using optogenetic stimulation in visual cortex in different brain states. And the question was whether, so how signals propagate in different brain states, right? Is it propagation, you know, we're assuming that, let's say, if we electrically stimulate the animal, you know, there's just improvement in behavior, or there's some things happening, and then no one is looking on trilateral variability of those results, and depending on the state of the animal. And what we found is that surprisingly, signal propagates much more, deeply during the state, during sleep states. So this is something that came to our surprise. We expect, if anything, given all these decades of work on stimulation, microstimulation, especially electrical, that will find these huge effects during, you know, the wakefulness, change behavior. But it's actually the opposite. We find that during wakefulness, effects are much more local. Whereas it could be that maybe you're targeting inhibitory cells postsynaptically, so it could be different possible answers. But during sleep, essentially, you have no barrier, no threshold, and signals just keep propagating because you have the synchronized up and down states that will promote that. But even if you control for that, you still find more propagation of signals using perturbation techniques in those sleep states, less aroused states. We also found that arousal plays a role in that. So in highly aroused states, you propagate less signals, which in a way makes sense because arousal is associated with attention. Attention acts on inhibitory mechanisms, which may block signals that are not deemed to be important. Yeah. Which also, you know, begs the question of how the different signals are treated, right? Signals that are important versus not important, are relevant for the animal or not.
Related to this, do you think attention and arousal is the same time scale?
Sorry?
Do you think that attention and arousal has the same time scale? Because I used to think that in this like brain state studies, like Tatiana's, for example, work, like attention is very short time scale now within the trial. Versus changes in arousal can be like super long, like minutes or even hours. No, so it can be like two different mechanisms causing the brain state changes.
But it depends. I think you can control arousal. So we have a line of experiments in the lab in which you're using this puff in the monkey's earlobe, and then we can, on a pretty rapid time scale, we can make the people go large when we do that, and versus small. So arousal could change on a more rapid time scale. So it's true with the reward, with expectation, may last, right, for seconds, or with locomotion, but in in different settings, you can control arousal and bring it at the same time scale as that of attention. But you're right, attention typically is faster time scale than arousal. Yeah, it could be also more localized, right? It's local effect. It's still propagate within networks, but it's not like whole brain, essentially. You're not just increasing responses throughout, it's just more specific. Yeah, so I think that we're now at the right time in the age of neuroscience with so many techniques and so many approaches that finally answer these questions, and many labs are aligning their work and trying to go in the same direction, collaborating. This is a fantastic time for neuroscience, really. I'm very excited. Yeah.
I also think it's like very difficult to, like, there is my background in mathematics, so I used to think like, it's hard to work when we don't have definitions. Like Anna started her talk, no? So for example, what is slow, what is fast, what is desynchronized, what is synchronized? Like it looks like we all study more or less the same phenomena, but like using different time scales and different special resolution. And therefore, there is no agreement, like, I believe, it's general comment, no, that whenever I try to talk about brain state, what I mean, it's more like transition between sleep versus wakefulness, like different stages of anesthesia, but some people may understand like it's always like between off and on transitions, like up and down state and stuff like this. And now on top of this, we have like during waking, we have different special propagation of this state. And I think that it's very difficult to place like all these puzzles in one big understanding what is going on, at least for me, I don't know.
Yeah, but I guess like ultimately phenomena on different spatial and temporal scales, they should converge to something. It's at least the way how I think about it. So today I presented you some dynamics we were studying on the mesoscale resolution, right? And it turns out it's very coordinated. It can be captured just with 6 dimensions. So this dynamics or this ongoing activity, as we see it with this type of mesoscale imaging, also has very striking features that it has bilateral symmetry, for example, right? Can we see it in single cells? For example, if you place 2 U2 arrays in different hemispheres and try to measure noise correlations, usually they would be very, very low across hemisphere, right? So it kind of seems like a paradox. How can we see such a synchronized activity at mesoscale, but it's hard to see on the level of individual neurons? So the way I think about it is that clearly like these signals present on mesoscales are also present in individual neurons, but they're just small part of single neuron activity. Single neurons are way more heterogeneous, way more higher dimensional than if we start to average them to get this aggregated signal. If we average enough of them, then we start to reveal these correlations which exist on the mesoscale, right? So I kind of think about those mesoscopic signals as presenting some notion of brain state influence the activity of neurons, but of course it's neurons in code. A visual notion of a local state when you start to zoom on local population. So I guess ultimately, like, we don't really disagree. Maybe we work on different, but if we start to look across scale, I think we should be able to find correspondence, or like the points where all those observations should meet.
But how do you, for example, explain, like, how would you put it? Because like this on-off transition in your work from are very local now, and attention modulates them, and so, and this is transition, like this is like the state when you have this on and off transitions. Traditionally was considered to be synchronized now, and on one hand you have this very local, but it should be also present globally, no? If it's really brain state transition related to synchronicity, shouldn't we observe it also like globally? I'm coming from like anesthetized animals perspective, so you know, this is the most global you can get. And then we go to wake, we see it, but nevertheless we don't see it globally, but we have global brain state fluctuation during wake. So what is the, I guess my confusion is like, we have this local on-off, which is synchronized. On top of this, we have study when we have waking different, like resting and waking quiet, waking active. And these are two different brain states. And at some point, I would like to place this on-off dynamics in one of those. And I cannot, because one is local and the other is global. Yeah, and so what is the, you know, what it would be the framework which could explain this both, like putting all together these both phenomena, I would say.
But I guess, like, when we ask like how global brain state influence dynamic of local circuits, they can influence timescales of the dynamics, but they can also influence the spatial scale of correlations, right? So if the global house, then these local, these synchronized states, I guess similar to what Valentin is showing, they can become more localized, and when we start to average over a larger area, we will not see synchrony, right? So it's also spatial extent of activity. So, and that's why I feel that if we consider both the spatial scale and the temporal scale, depending on which of those scales we do define synchrony or time scales, there are common points of correspondence. So going back to this question of local on-off dynamics, my view is that, let's say when arousal is getting lower, the animals get less engaged, then this dynamics becomes more synchronized across space. I guess it's also similar to what Valentin has mentioned, and during sleep, you would have an extreme of this where entire cortex can engage in slow oscillation and signals can propagate very, very globally. So it's also the spatial scale of this synchrony, which I guess matters and changes with the global state.
Yeah, I also agree with Tatiana. What we see in our models is, for example, in what I presented, that if we look at the very non-synchronized type of activities, stochastic transitions, and if we look at it locally, we see one picture. If we just pull the data without averaging, but if we pull it together kind of in a like summing up, as some of the global measures do, we would not see anything anymore. We would not see the signatures of connectivity in the activity, and we would have a completely different idea about timescales and what is important locally. So there is some kind of, like, there is a good averaging that highlights important parts, and there is an average that, like, basically the signatures that getting continuously kind of making more visible by averaging are possibly the ones that are indeed persisting across scales, and some are local, and then there would be at least at some point decrease in their visibility, because by averaging we average once it's synchronized to one phase and one synchronized to another phase, we don't see anything anymore. Something like this. Like there is kind of this maybe also a window into understanding how local things are by coarse graining. But for this we need to have a lot of very well resolved signal also, maybe then.
I always have this image of the brain fluctuating like the ocean, and when we do task, these operations are like small boats within these resting state fluctuations that are dominating. Because when you see data, well, as you see, Tatiana, when you see the single trials, it looks like there is some patterns there, and you barely can see a signature of the stimulus, for example. But the stimulus is perturbing us. It's like overwhelmed in these fluctuations. So yeah, I always have this image of ocean with small boats that represent our perceptions and maybe our actions and motor commands and so on.
But if it's like ocean, then we have like global waves, no? Of yeah, like slow transition in the brain states, which are super globally synchronized. Right?
Also, because and this is also maybe it relates to Tatiana's work. We often have models of spontaneous activity which are at the stationary regime and in a closed system, which is problematic, I think, because the system, the brain is an open system that is interacting all the time with the environment, no? And it's out of equilibrium. Otherwise, it would be that. And so, but maybe what we're seeing, and that is correlated with all the behavior of the animal, of all the movement of the rats, as it's correlated with the video of the experiment. It's telling you that the system is it's really exchanging energy with the environment all the time. But in our models, we always assume that it's intrinsic activity, self-generated by the network, and that reverberates there, and it's like isolated from the environment. So there is the missing part that is the signals from the organs. There is no such a thing like a resting state, pure resting state. So there is always like signals coming from the organs, from the at least from the organs of the environment.
But oh, go ahead. Sorry.
No, it's okay. Good.
Valentin, please go ahead.
No, so I was going to say that is a resting state. I mean, all this like dancing, right, that happens at this micro scale, it is a resting state. It's what we know as a resting state. But I have not seen any modeling work actually capturing this resting state dynamics, and then how these different states could emerge in real time, right? Have you seen any any computational work, like looking at this interaction between local and more global states and how they can coexist? I don't think modeling is able to reconcile this. Yeah. What kind of mechanism could? I mean, the simplest mechanism is that you go in synchronized state and then go to synaptic depression, and then response go down, and then.
That's my paper.
Yeah, I know, I understand, but exactly. But it's more than that, right? This is one way to look at it, but then, I mean, it doesn't explain all this interaction with the more global states, right? Synaptic.
But I guess the modeling work will go there, right? So there are good models of brain states which don't have any spatial structure, which don't really have realistic connectivity of the entire brain, for example. But we do have data sets about connectivity now. So I think it's just a matter of time until these biophysical models, which very precisely capture, let's say, sleep-wake transitions just in unstructured network, until this model will appear, well, where they are put on a realistic connectivity, and then we can study emergence of states as we observe them in animals, where there is a notion of large-scale activity, which have very specific characteristics and how it influences local circuits. I agree, I don't think I saw anything of that kind existing, but I think we should see it soon, I would guess.
Yeah, so maybe, I mean, one reason maybe why the states exist is to preserve the integrity of the information that is maybe locally stored in the network, right? You don't want that information to go away, so you want to be some sort of maintenance of information. Then it's question what happens in a longer time scale when you want to maybe consolidate information or transfer to different systems. So what kind of dynamics would explain that? I feel like we're only scratching the surface of this. This is what I want to say, Gabriela. It's not like, yeah, we have this understanding, mechanistic understanding at a local scale how this on-off could emerge, but then, you know, to reconcile this with the more global states and longer time dynamics, and are they meaningful in any way? Do they mean something for the organism? Are they needed, or just an epiphenomenon?
I was actually thinking about this throughout your presentation, especially coming from the perspective of energy. It seems to be very energetically costly to have this synchronized state for the system. And I would expect that coming from a low energy perspective, the system would try to minimize the expenditure of energy of going to synchronized states. So, for example, one of the perspectives that I was speaking about is whether there is a good balance of the amount or the amount of time that you spend in a synchronized state compared to a desynchronized state. And if in an unhealthy brain, that balance would be damaged. And then you could also test that maybe with performance, but if, as I think you proposed, I mean, I'm not familiar with the paper, so I might have misunderstood, but the desynchronized state seems to be more efficient for the performance of the animal in that particular example. So why would a synchronized state that doesn't seem to be as efficient exist? I think for me, it's like one of the most interesting questions. Why do we need that? And if we need that, how much do we need? Because we don't want to go into that state all the time, because then we wouldn't be able to perform as well. So
actually, I think you'll be surprised that the desynchronized state is more metabolically costly, right? Because firing rates are higher in desynchronized state. So if anything, synchronized is the default mode of cortical networks. So if you go to like in vitro and just have a network of neurons, they'll oscillate, right? This is the default mode of any network, to oscillate. This is the resting state, right? Just go up and up and up and on. This is what they do. Now, only when the stimulus comes, when the animal is active, the state becomes desynchronized, and that is when it's become costly. And I'm sure we all know that resting state takes most of the metabolic expense of the brain, like 85% of amount of oxygen is consumed during this resting state or spontaneous activity of the neuron. Only 15% is accounted by the task, by the animal being active, right? So they must be doing something, right? Because the brain is always in that state that is, you know, the preparedness for what's coming next. And it seems like it's something very important. Or it's doing something that we're missing completely. Yeah. But you have to think about, I mean, when you talk about synchronized states, you're not necessarily thinking locally synchronized. Because yeah, you're right. I mean, you have a lot of surgeons firing, but that cannot resist because like Gabriela says, synapses will depress and then force network in a in a lower state, in up and down state, right? So to speak. But this synchronized state is actually associated with lower firing of neurons. So if anything, it's not that costly because it's kind of it's easy, right? Synapses depress and go down and up and down, etc. Then inhibitor neurons are interacting with this. This is the entire network effect. But to maintain the network in desynchronized state, this is what consumes energy.
Okay, that's very interesting, but I think we should we should finish the workshop because we are a bit late. Thank you very much to the speakers. It was great to having you, and I hope we continue being in touch, and that we can meet in person. We can meet in person one of these days, and not only online.
Within Paris, for good.
Yeah.
Thank you,
Thank you
Gabriela.
very much for everyone. It was a pleasure to have you.
That was great.
Thank you so much for organizing this. It was a lot of fun. Thank you.
Thank you.
Yeah.